Hello, good morning, good afternoon, and a good start of your evening. Thank you very much for joining the second day of the Animal-Free Safety Assessment Collaboration workshop on the transitioning of tetanus, diphtheria, and pertussis-containing vaccines to animal-free batch release testing strategy. The focus of today will be on the potency testing. So just very briefly, some information about how to interact with us. So this is a webinar, so the participants will have their video and audio automatically muted. Please, if you want to interact with us, use the chat in Teams. So we will monitor the chat. If you have problems with the chat, there is also the Slido that you can use where you can put your comments and questions over there. We will try to monitor all of this. Again, if you can, make sure to write down who you are when you are interacting with us, because it's important for us to understand what is your background and from which organization you're coming from. So again, today, as I said, we really hope to get into the details about the potency opportunities for this type of vaccines. Those are vaccines that are heavily relying on the animal testing, and the numbers of animals used for the potency test of tetanus, diphtheria, and pertussis-containing vaccines is probably one of the highest within the batch release strategy context, together, of course, with the rabies vaccine that is also another one that is demanding a lot. So for today, we really hope to give you a good idea about what kind of opportunities are available. And as yesterday, we would like to share with you the knowledge about the available opportunity, what has been developed, what is currently being used by some of the speakers that are going to present today. We would like to hear from you about your experience, if you have any direct experience in trying out some of those, what kind of complexity you might have experienced or you fear to experience when approaching this type of methodologies. And of course, what is also important for us is having a conversation about how to facilitate the implementation, not only from the batch release approach, but also in terms of updating the current pharmacopoeia or regulations worldwide, with an idea to make sure to make this transformation something that is as aligned as possible everywhere in the world. The agenda for today will start with an overview about the use of the single dilution. Then we will get a little bit more in the details about the full in vitro potency test. You will hear the methodology that has been developed for tetanus, diphtheria, and acellular pertussis. We will hear about the perspective of the manufacturers from the developing countries in approaching some of those methodologies. And then we will get a little bit into the details about the wholesale pertussis potency test, which is again probably one of the latest challenges that we need to solve. And as yesterday, we will also have the chance to have a nice panel discussion where we really hope to hear from you. So again, without further ado, I think that we can definitely start to hear from the speakers, and thank you very much again for being here with us today. Thank you.
Yes, and with this, we start into the first topic of today. As you know, there's the multi-dilution assay, but there's another possibility, the single dilution assay. And therefore, I ask Dr. Pradeep Kumar Das to join in to give his presentation on this, what experience he has made with this topic. And Dr. Pradeep Kumar Das, he is the General Manager of the QC department of the company Biological E Limited, based in India. Please, Pradeep, start your presentation.
Thank you, Cynthia, and thank you, Laura, for once again giving me the opportunity to share my knowledge and experience on how I derived the single dilution assay for diphtheria and tetanus component. Let me share my screen. Can you able to see my screen?
Yes, we can see it.
Yeah, thank you. So single dilution assay is mainly for diphtheria and tetanus component and its combination vaccines, whatever the available. So as you know, for most of the things, multi-dilution is the common approach for routine batch release, but there are certain additional tests are available as per the compendia conditions where the single dilution also can be used for routine batch release. So these conditions are very well defined in WHO IVB 11.11, where the basic information is needed is the condition was first condition is evidence of consistency in production and testing, evidence for highly significant regression of dose response lines for vaccines and justification and assumptions, guidance for the selection of single dilution assay parameters, and prediction of behavior of single dilution system. So in this, manufacturer who has prior experience of a multi-dilution assay can go for a single dilution assay by practicing basically 10 to 20 recent or consecutive multi-dilution assay. So this is very much important when you are optimizing your single dilution assay. So this is the minimum requirement. But as you know, the animals, the test, it is much of a variable. So more data, if it is available, more the variability it is getting reduced. So it is very much important as much as data multi-dilution is available, you should consider for your consider for single dilution for routine batch release. So there are certain objectives are also for the single dilution, as you mentioned. Any change in the vaccine production process, that is your purification step, adjuvant formulation, or testing methodology, should have evidence must be provided. So any change in any of these conditions, you need to perform again a multi-dilution and show the consistency approach. So this is again, I have compiling from the various TRS and reference document what the each document says. So as per WHO TRS 980, the one dilution can be of same principle for evaluating the response for three dilution assay, and the statistical response should be the reference vaccine should be P should be less than 0.05 for the considering potency for the test vaccine is satisfactory. So this is the this is the numbers derived by a statistical data where your batch release should be, or your product should be less than 0.05. So one of the disadvantage of this single dilution is also there, that it is strictly quantitative estimates of the vaccine potency. Hence, when you are when the manufacturers or those who are doing this assay, they have to very critically critique the methodology and the results what have been obtained during the multi-dilution approach. If that approach is good, then only this single dilution assay work. The whole single dilution assay is basically on statistics and the derived single dilution. So as we know, the multi-dilution criteria, the p-value should be less than 0.05 for the significant statistical analysis regression curve, and for linearity and parallelism, it should p is equal to greater than 0.05. So I am just beginning just how we have derived the data. There is another chapter that is an old document, that is VSK 97.04 chapter, where it obtains how to perform the assay. I have taken 29 batches data, consecutive batches data, and I have obtained the mean value of relative potency, LCL value, and UCL value. So these three are first, and you should get the percentage CV, standard deviation, and mean. Once that is identified as per the guidance which I have shown in my earlier slide, so the protection should be greater than 20 to 20%. So when you are identifying, okay, this is my dilution at the least, which is coming as 0.77 for the relative percentage confidence interval. So as the confidence interval is the less than 20%, it is acceptable for the in vivo testing. So these three criteria are very much important for defining the consistency of the production and testing for the diphtheria component. So first, a parameter is the mean potency, and you have identified mean, standard deviation, and percentage CV. So next criteria is evidence of highly significant regression and justification for assumptions for linearity and parallelism of dose response curve. So when we have taken 29 sets of data, which is the reference standards we have taken out for the regression, non-parallelism and non-linearity. This you can calculate by using CombiStat software. And in this software, you can get all these parameters like regression parameters, non-parallelism, non-linearity. So your value should be, probability value should be less than 0.05 is significant. If it is less than 0.01, it is highly significant. And if it is 0.001 is more significant. So this means that the product has the consistency and it has a significant parallelism and linearity between the dose curve from reference and test vaccine. From this table, we conclude that our data is highly significant regression from the dose response and no significant difference observed between linearity and parallelism against the reference standard. So third important parameter is guidance for selection of single dilution system parameters, namely the number of animals and dilution level to be used in reference vaccine. So my reference vaccine is diluted is 40 IU per ml. I am diluting it to 1/10, 20, 40. And here it comes the survival. So this is the average survival. And what is the percentage survival we are getting? So I got with this dilution, my percentage survival is 6.57 in 1/40. Hence, considering this as per the guidance, the 10 to 20% of protection is considered to be acceptable. This means the protection level is below. Hence, I have considered 1/40 dilution, which is about 6.57 is the percentage. I have taken one more additional dilution of 1/20 to meet the percentage survival criteria. So minimum your dilution should be selected to 10 to 20% protection level should be considered for your reference standard. Once you are identifying this, you calculate the ED10 effective dose, 10 dose. So you need to identify at what dose level you are getting the reference standard for the effective dose parameter. So out of 29 samples, I have 15 times I have used the reference standard. I have taken the effective dose at 10. So what is the 10? The mean average dose is 37.95 is the calculation parameter. And the 10% protections comes to 1.05. So if you see the guidance for the reference vaccine, historical data used to select dilutions containing the number of international units that exceeds the immune response for all quantal response, 10 to 20% protection is considered acceptable. So when you are calculating your reference, the potency of the reference standard is 40 IU per ml and dose required to at least 10% protection is 1.05 ml. So I have to do to obtain this dilution, how much to IU per ml? My dilution of the reference standard should be around 38-fold. So this dilution of fold is we need to do when you are obtaining from the single dilution. When you obtain the reference standard single dilution, further test need to be carried out. Now, selection of dilution for the test vaccine. So what is our test vaccine requirement for diphtheria component? As the guidance say, it should be 30 IU per single human dose per vaccine. And in the WHO 97.04, to verify that single dilution method likely to work and compare response for the lower dilution reference of the reference vaccines, which should be chosen to elect minimal response. So from this data, what we have arrived that the reference standard is diluted to 38-fold, and to obtain 38-fold and minimum IU concentration is 30 IU per ml. And based on this, as we obtained 1.05 ml, we got approximately 57.14-fold approx, which is around 57-fold for the test vaccine. So now you have arrived for both reference vaccine single dilution and the test vaccine single dilution. The next criteria is, which is the foremost criteria, is how much number of animals I should use for single dilution assay. So in chapter VSQ 97.04, so it has been obtained that once you are the minimum potency factors, so which has to be calculated from a slope, that is the mean slope, And with this 15 data, you are obtaining the mean slope of 3.37, which is approximately 3. So 3 slope, how much number of animals I should use? I can use 10 also, 15 also, 20. But basically to begin for a beginners, my data is coming with 3. So I should use 20 number of animals as per this slope parameter. That is the minimum criteria which will give us as a prediction with the slope number 3, if we use 20 animals for single dilution, we have to contain 30 into 3.4, 97.20 IU per SSD for the diphtheria potency. So once you calculate this, there is another requirement. How is the prediction of behavior of single dilution assay? So is the survival of the reference vaccine, you give the survival of 1/40, what you obtained, and survival of the test vaccine. And what is your p-value? Based on your p-value, if it is less than 0.05, it should be significant. So this is derived from the Fisher extract and also the statistical calculation which you can use with the Combistat. So calculation of the test vaccine has shown significantly higher response than the reference vaccine, which says that the batch meets the minimum potency requirement. So once you derive this, you need to perform the validations of the single dilution assay. You take 3 single dilution tests simultaneously and perform the acceptance criteria. So your 3 batches simultaneously, once you do the analysis, the probability value should be 0.05 and it should be less than 0.01 or 0.001, even more significant. And if a batch fail in the single dilution, you need to repeat the assay with multi-dilution assay. So that is a basic requirement. So this is a hypothesis which we have done with the validations. So we have gone with 1/38 dilution for the reference vaccine, and for the test vaccine is 1/57. So this is the probability values when we perform. We have taken last time point stability samples to understand if there is a degradation. Also, we have routine batches also we have tested by doing this hypothesis, which indicate that the test single dilution assays meet the criteria of probability, which is less than 0.05 for dilution. Hence, the single dilution assay may be implemented, and after approval of NRA, the diphtheria potency can be used for routine combination vaccines. So these are the conditions for single dilution assay. So if as I mentioned in the earlier slide, the conditions, if anything, there is a significant change in the production process, testing should revert to multi-dilution assay. And to monitor production consistency, full multi-dilution assay should be performed and it should be approved by NRA. If there is a change in reference standard, minimum 3 independent multi-dilution assays should be performed to identify suitable single dilution by statistical analysis. If the batch fails to meet P less than 0.05, the sample should be retested and followed by multi-dilution assay to decide the fate of the batch. So this basically the conditions which shows the presentation. Similarly, I did for the tetanus component, but I will not go in detail for this. But I once again thank you for giving me the opportunity. And if any questions are there, please do let me know. Thank you.
Thank you very much, Pradeep, for this very, very interesting presentation. I checked the box. Currently, there are no any questions. The only question, maybe I can ask a question. You said when the single dilution assay fails, then of course the multi-dilution assay has to be done. I'm wondering how often this happens and how you keep people, your lab people, trained in a multi-dilution assay, because you have to use more animals, different dilutions. So how do you overcome maybe this challenge?
So yeah, right. Good question. So basically, single dilution cannot be just directly with a initial stages. So people who are working in the manufacturing team or the testing team, should be very well experienced. They should have understanding on multi-dilution assay. And once you derive maximum batches, so I have taken 30 batches data, but people can take as much as data which is available, 50 batches, The more data you will take, more accuracy of your research. And as I mentioned, single dilution is purely statistically derived product. If your dilutions are incorrect, or you have a different issues in identifying the things, your single dilution will not work. So in your lab, the data, whatever you are obtaining from the multi-dilution assay, the statistical significance for all the parameters is should be looked into. So as this assay is quantitative, so based on the NRA, once you give the data to the NRA, so NRA will suggest that every 1/15 batch you need to perform the single dilution, or based on your historical data, they may increase to 15 to 30 or further. So this is the main challenge. You need to have this, but it definitely reduces the animal consumption by around 60 to 70%.
Thank you very much for this clarification. And probably also another challenge is, okay, the single dilution assay failed. Now you have to order again animal for the multi-dilution assay. So this really elongs the time period until you're really ready to release your batch. What is your experience with this? Is the animal delivering companies, are they quick? Do we have to wait sometimes until the animals are delivered for the multi-dilution assay?
So most of the animal houses have their own breeding facility to meet the production requirement, or they outsource also to the laboratory, and they bring the animals based on the planning. So it requires minimum of 3 months of planning of the what are the batches coming in upcoming 3 months. So it has a very long process, pipelines, and the animals availability. It is definitely a very challenging thing. Multi-dilution assay may not be a significant where if you want to go for higher production. So this is, we have attended for a 3 year. This has at least have a minimum 60 to 70% reduction. But going forward in upcoming a decade, in next 10 years, we should move out from this method to a purely a in vitro based method. That is the ultimate aim.
Very fantastic goal. Thank you so much.
Thank you. Thank you.
Thank you, Pradeep, and thank you, Marie. So I'm actually happy to call up for the next speaker that is also going to provide some insights about the pros and cons of the single dilution assay. So it's my pleasure to welcome Morgan Florence from Scienzano, from the Department of the Quality of Vaccines and Blood Products. So Morgan, thank you very much for being with us today. The floor is yours.
Thank you, Laura. I'm going to start by sharing my screen. All right. Can you see my screen?
Yes, not yet in the presentation mode though.
Yeah.
Perfect. All good. Thank you.
Thank you. Yeah, so I'm going to probably repeat a few things Pradeep already mentioned. I'm going to give you like the overview from our side as the Belgian OMCL. I wanted to start by giving a little bit of background. So although I'm sure most of you know what DTP vaccines are, but just to give a bit of context, about why it made sense to implement a single dilution challenge assay at some points, how to implement it in practice, conditions, and again, pros and cons, so advantages and limitations, and then move on just to introduce some other perspectives that we can work with. So as probably you know, DTP vaccines, they are like quite old vaccines. They were developed in the 1930s, authorized for market in the 50s. Which means that back then the 3 R's were not so much of a concern. And for potency assessments, the assays that were implemented were multiple dilution challenge assays. And up to fairly recently, there were limited alternatives to these assays. So what we do in a challenge assay basically is the animals which are guinea pigs for diphtheria, mice for tetanus, are injected with a reference vaccine or a tested vaccine, depending on the group. And then 28 days later, we have a lethal challenge with an injection of the toxin solution. And then the animals go on developing symptoms or not, and ultimately dying in some cases or not. So when the 3 Rs became more of a concern, we started to implement humane endpoints also, so that we did not have to wait for the animals to die, but we could euthanize them a bit earlier than that. For example, with the complete paralysis of the hind leg in the case of the mice treated with the tetanus vaccine. So that was already a way of implementing the 3 Rs somehow. But you have to realize that these assays demand a lot of animals. We have to count 4 dilutions per reference, 4 dilutions per tested vaccine, with each time 12 to 16 animals per dilution. You have several controls, toxin activity control, challenge dose control, with again, several dilutions. And so basically in this case, we compare the number of survivors in each dilution of both group, reference vaccine, tested vaccine, to establish the potency. So now, when the 3 Rs became more or gained more importance, the WHO started to encourage to reduce the use of lab animals, also via the EU Directive 2010/63 EU. So the idea is to target a reduction in the use of lab animals and also less animal suffering. So about guidelines, when can we use a single dilution challenge assay? So basically, this is a bit of a repetition from what Pradeep said. This has to be done for a specific product, showing consistency in production and testing. You first need to have adequate experience with a multiple dilution assay for this product, and you need to perform a validation for your single dilution assay. So a bit more in details, what does that mean? For the adequate experience with an MDA, you need very good hands-on experience with the assay. Again, product specific. You need to have a validated method in terms of repeatability, reproducibility, robustness, etc., different criteria. And you need to have a large base of good data consistency. If you have that, you can move on to your SDA validation. I'm just summarizing here. Pradeep went really well into detail with this, but starting from your set of consistent data with your MDA, you start by the determination of your reference dilution, so reference vaccine dilution to confer like 10 to 20% protection in the reference group. Then you move on to calculating your vaccine dilution. So based on, you need to choose the minimum dilution required compared to the dilution you choose for the reference. And then you can start predicting on the behavior of your SDA, and you need to have a maximum of 10% discrepancy between your actual results with the MDA and your prediction for the SDA. If you have all that, then you can start applying your SDA for actual testing. This made a huge improvement because it allowed us to move from 3 to 4 dilution per reference and vaccine to only 1. Also for the controls, a reduction in the number of animals used for controls, because instead of being for each test, we could do them like a couple of controls a year. Of course, this changes the outcome of the test. And as Pradeep said, this maybe is already a limitation of this single dilution assay, is that instead of having your potency in IU per dose, like an actual quantification, you have like more of a binary pass or fail result. So it's a little bit less informative, but this allows for a decrease of the number of animals by up to 80% with all the controls and everything. Other advantages we may mention is that you can have a higher number of vaccines tested in one run. It's definitely a reduction of costs and resources. Also in terms of space, like less space required in the animal facilities. And it definitely also saves time for the operators and the animal caretakers, of course, because they are fewer animals. So limitations, as I said, the necessity to have first an adequate experience of with the MDA. Also maybe the binary outcome, pass or fail. And in my opinion, definitely it is still an in vivo method. So it is still using lab animals. It is still a challenge assay, so involving a lot of animal suffering. We wait for the animals to develop symptoms of the disease and eventually die. So we thought there was still a lot of room for improvement there. So already starting with like other perspectives, in our lab, we decided to move to DTP serology. So in this case, as we do for the pertussis components already, instead of the challenge assay, we have mice that are injected with the tested versus reference vaccine. On day 0, and 28 days later, the animals are anesthetized, bled, and we collect the sera to perform ELISAs, and we measure the potency in terms of antibody titers for diphtheria, tetanus, and pertussis. So in this case, we are mostly addressing animal suffering because we do not have to wait for animals to become sick anymore. Not so much in terms of animal number reduction. I'm going to explain why. So this, of course, also was a step-by-step process, starting from our challenge assays that were performed, if you remember, in guinea pigs for diphtheria, tetanus was in mice, and then on the side we had our separate set of mice for the pertussis serology. Then we moved to one step where we had diphtheria and tetanus serology implemented in one group of mice and still our pertussis serology on the side. And eventually we moved to a DTP serology in one single group of mice, but to make our assay more solid, the DT and DTP serology were developed in a multiple dilution setup. So this is definitely better in terms of animal suffering, but we have to, again, move to multiple dilution. So in terms of numbers, we decreased from, this is an example for like the test of two test vaccines. So we moved from 363 animals for the challenge assays and pertussis serology to 107 animals for two vaccines tested when we were in a single dilution setup. Then moving to the DTP serology with a multiple dilution setup, we are back to 135 animals in total. But ultimately, the goal now is to move to single dose DTP, so serology. And then we end up with a total number of 45 animals. So that would be also a drastic reduction in the number of animals. So addressing both numbers and suffering. Of course, I do think this is not the ultimate response to the testing of diphtheria and tetanus potency. I think in vitro alternatives will always be like the best ultimate choice for being animal-free, for saving even more time and costs, for the possibility to assess all antigens in one run, and also because they show much less variability than in vivo methods. So I'm thinking about multiplex assays, ELISA, but I will let the next speakers this morning go more into details for this. So just, one conclusion slide to recap the pros and cons. Pros of the SDA, again, drastic reduction in numbers, more vaccines tested in one run, reduction of time, costs, and resources, and less space required. Limitation, the binary readout, pass-fail. And again, it's still an in vivo method and a toxin challenge, so room for improvements. Yeah, that's it for me. I don't know if you want to ask questions now or if we move on. Thank you for your attention.
Thank you, Morgan. Just very quickly, because I think that it might be interesting to learn a little bit more about the process that you are putting in place to move to the drastic reduction. So having one serology, single dilution for tetanus, diphtheria, and cellular pertussis. So how long did it take to really go through the process? How much confidence you needed to have within the process in order to be 100% sure that this is your current strategy for the release of the products? If you can say some words about that, might be interesting.
Well, again, the first thing is that to be sure that our assay was like, to be confident enough with the process, we had to move back to multiple dilution assay because that was more informative, more precise. And then only in a second like step, or in a third step even, we will move to single dilution. So that's one first like downside, or like intermediary step. The otherwise for the process, I would say it took a bit less than a year, but we had to definitely compare like our results. We started to implement the serology on vaccines that had already been tested and approved with or single dilution or even multiple dilution processes, and really by comparing both and building enough data consistency to become confident with our serology in a multiple dilution. So ideally we try to, we even reperformed some multiple dilution assays just to be sure to have ways to compare like multiple and multiple together. So, and to have, and again, product specific. So we had to do it like separately for all the vaccines that we have in our pipe. So, yeah.
Margen, may I also ask you one question? As we are part of the OMCL network in Europe, there also would have been the possibility to implement the reduced testing scheme, you know.
We are doing this. Sorry, I should have mentioned it. We are definitely doing that. Yeah.
Okay, you're doing this.
Yeah,
okay.
We kept our reduction scheme. Yeah.
Okay.
So we were already using that with the challenge assay. Sorry, I interrupted you. I don't know if this was the question, but we are definitely doing that.
Okay, this was just the comment from my side, and I'm very happy to hear this.
Sure.
Thank you so much. Thank you so much.
Morgan, there are a couple of questions in the chat. I don't know if you would like to have a look and take it now or later. It's about the reference standard for the diphtheria toxoid, and if you can share also some information about the working serum standard used for DT and the cellular pertussis antigen estimation.
So the reference, we are still using the BRP 3 and 4. And can you please provide? This I cannot say. Like we work in 2 teams, and this is like basically the ELISA team that has the specific info if you want to. But I can ask and come back to you.
Mm-hmm.
I also have a question here. It comes from Gabriela Radulova. A question based on the slide with ELISA assay. In the brackets, you wrote that the potency can be shortened to 1 day with ELISA, not 30 days, but the animal will take at least 28 days to develop antibodies, which are needed for the ELISA. Can you please elaborate on this?
You mean with the serology? Because no, I think, but this will be, or with the last slides that it moved, but then I think I'm not the one to have to elaborate on this. I think it will be the job of Paul later on.
Okay.
I don't want to spoil the what's coming.
Okay, then this will be a question then for Paul Stickings afterwards.
Thank you, Morgana, and thank you.
Thank you so much. Okay. Then I would say we will then move on to the next speaker. It is Silvio Bandiera. He's working with Sanofi. Hello, Silvio. We are very happy to listen now to you. What is your experience on this topic?
Yes. So good morning, everyone, and thanks for inviting me today to present about SIA. So first of all, just logistics. Can you see my screen?
Yes, we can see your screen and you too.
Perfect. Excellent. So I'm what you internally call the analytical product leader within Sanofi Vaccines, and I'm specifically dedicated to the AXIM family products, top pediatric combination vaccine, DTwP backboned pediatric combination vaccines. So the role of the analytical product leader within the company is to, let's say, lead and drive and coordinate the changes in the product control profile versus new standards. And one of these new standards, which is very important for both ethics and industrial performance, is the 3Rs initiative. So trying to refine, reduce, and finally replace animal use for potency testing. So again, thank you. I will talk today about the DTwP single immunogenicity assay as applied to AXIM vaccines for routine release and stability testing. So the AXIM brand basically includes three main products, three main vaccines. Tetraxim is our tetravalent vaccine, anti-diphtheria, tetanus, acellular pertussis, two purified components, pertussis toxoid and FHA, and poliomyelitis. Tetraxim combined to our lyophilized Haemophilus influenza vaccine active gives Pentaxim, the pentavalent vaccine, and then there is Hexaxim. This is the fully liquid hexavalent vaccines, which two previous balances had the recombinant hepatitis B surface antigen produced in yeast. In this slide, I try to schematize the change we proceeded into, starting from traditional methods for testing DT, PT, and FHA potency to SIA, to the single immunogenicity assay in guinea pig. All methods are based on compendial, European Pharmacopoeia compendial methods. As you can see, traditional method for diphtheria and tetanus use toxin challenge assays, intradermal reaction challenge test for diphtheria, and paralysis induction challenge test for tetanus. And for PT and FHA, for pertussis, we had already a serological method in mouse, as you will see, so already based on a single dose approach. With SIA, basically, we moved to a single immunization model, as actually suggested by the European Pharmacopoeia in case of combo vaccines for which the DTwP potency has to be tested. And we also combined that guinea pig serology with multiplex antibody quantitation by Luminex technology. So the quantification of different antibodies for different valences for the different antigens in the very same serum sample. And we based actually the decision to make this change on the specific regulatory frame we were into. We had the specific chapter of the pertussis potency testing, which clearly indicated that in case of combination vaccine, the serology assay in guinea pig should be performed in the same group of animals. And also, of course, we had the European Directive 2010, which Morgan just mentioned, which is one of the most stringent ethical and welfare standards for the protection of animals for laboratory use. So matching the previous table, in this case, we want actually to reproduce how the reportable value changed by moving from traditional method to SIA, and how also acceptance, specification limits, if you prefer, had to be set. So previous traditional methods for diphtheria and tetanus had a multiple dilution design, as you have just seen, where 4 doses of a reference vaccine titer, which has an international unit per single human dose titer, are plotted against the dose response profile of a sample vaccine in blue here. And by calculating, of course, the relative potency, so the relative shift between the two dose response profiles, we can assign to the sample vaccine titer a international units per single human dose titer and check these versus official acceptance limits. Same thing for tetanus. So we have always mean clinical signs, let's say, which reduces over time versus the increase of the vaccine dose. For serology, the design was already a single dose approach, no international unitage existing for pertussis, PT, and FHA. So the approach was a relative potency approach as well, but with an acceptance limits for the sample vaccine antibody titer not to be significantly inferior to the reference vaccine titer. With a single immunogenicity assay, we introduced a new way to generate an acceptance limits for DTwP immunogenicity, which is specific to this product, which has to be specific for this product and to the consistency of manufacturing of this product. The calculation of that limit is to take at least for a preliminary acceptance limits at least 15 mean serum antibody titers at a specific given dose dilution of the vaccine. Calculate first, verify of course the normality of distribution of the data, calculate the mean, and set the acceptance limits at the what we call the control limit, basically, of the manufacturing process, which is the mean minus 3 standard deviation of the mean. But before to possibly do that, has been introduced before to possibly move to a single dilution approach, we had to set a full and comprehensive suitability study built on a multiple dilution design to possibly choose that single dilution and then move to this consistency approach for setting specifications. So what was this step-by-step process? What was the step-by-step process we applied for SIA that has been approved by regulatory authorities in Europe and worldwide? So first, we had to discuss with health authorities and obtain endorsement for applying to diphtheria and tetanus the serological approach that is described in the chapter 2716 for acellular pertussis serology in guinea pig, which is, as has been presented before, once suitable dilutions have been confirmed for a given vaccine, then try to move to the single dilution approach. And when we move to a single dilution approach, choose between two possible reportable values, either the ratio of the geometric mean and titer between the sample vaccine and a reference vaccine or an internal control vaccine, or directly move to the geometric mean titers of the antibodies induced by the test vaccine, which is the consistency approach, which you actually choose at the end. Then once that, let's say, transition of the principle of serology expressed for pertussis could have been, let's say, applied to diphtheria and tetanus, then first of all, we had, of course, to perform a CHQ to compliant full validation of what we call the in vitro part of the test, which is the quantitation of anti-DT and AP antibodies, simultaneous quantitation by the Luminex technology. So of course, we had to look for specificity, linearity, accuracy, and precision for each antigen, antibody quantitation, and that made in 2019 the object of a publication I referenced here for your interest, of course, if you want to access that publication. Then we had to perform this comprehensive in vivo suitability study using a multiple dilution design as per the European Pharmacopoeia indications. So first of all, we had to calibrate, to assign international units per single human dose titer to our internal reference standard vaccine. The internal reference standard vaccine we want to use for the single immunogenicity assay. So that means run at least 15 independent tests on previous traditional challenge methods in order versus official reference standards in order to gather titer international units per single human dose for our SIA internal reference standard. Then perform multiple dilution testing in double, of course, traditional methods and SIA, using the internal reference standard vaccine in SIA on at least 15 commercial batches, possibly including batches that give the proof of clinical efficacy, in order to first, as introduced before by Pradip, verify linear and parallel dose response profile on at least 3 consecutive dilutions. The criteria applied for linearity and parallelism are exactly those that Pradip presented before. Verify that the calculated international units per single human dose titers obtained by SIA meet traditional methods acceptance limits, which is SIA at least as discriminatory in international units per single human dose as traditional DNT challenge methods. And then only once these criteria were met, choose a dilution in the linear and parallel SIA dose response range for proceeding in the single dilution approach. Then on the same, of course, set of batches that showed linear and parallel profiles, established these consistency-based preliminary acceptance limits on the antibody titers method at the specific chosen dilution, which in our case was the highest concentration, so the lowest dilution in the validated dose response range. And last but certainly not least, plan and execute what we call the SIA, the method life cycle management, which means reevaluate the appropriateness of the established preliminary acceptance limits on at least 15 commercial batches when more release data are available. Here is an example, at least 100 batch available for reevaluate the appropriateness of those criteria, and also periodically conduct SIA multiple dilution testing in order to continuously confirm the validated range that has been performed during the suitability, that's been obtained during the suitability study, and also run SIA multiple dilution testing when comparability data have to be provided for major manufacturing process changes of our vaccines. So between 2020 and 2022, we gathered approval for the 2 products, Tetraxim and Hexaxim, in Europe and worldwide, for moving from traditional methods to SIA single dose methods. You have again here an idea of the reduction of number of animals used when comparing traditional methods and new methods. Here there are estimation per batch. And basically, now we can really see we have, hands-on experience on the advantages that these changes brought, moving from 3 methods to 1 methods, extremely significant reduction in animal use. We estimate in 3 years for Hexaxim only a reduction of 105,000 animals less used for laboratory testing. No suffering, limited sufferings, meaning that we refined the test so there is no longer toxin challenge of the animals. We also reduced invalidity percentage rates. We reduced testing time, and therefore we reduced the risk in discontinuity of supply, which was quite high with the previously adopted methods. So this is the present. What's next? So this is, I think, a link with the next session. The future will be to get fully acquainted of animal use when sufficient comparability data, of course, and design will be worked on for this antigenicity license. So the future will be to completely replace animal use and move to antigenicity testing for replacing in vivo potency testing. And so these are key colleagues, let's say, who participated to the success story of SIA. I really want your knowledge, and really thank you for your attention. I'll be happy to answer to any question, please.
Thank you very much, Silvio, for your presentation. Currently, there aren't any questions in the chat. Maybe I just come back to slide number 7, where you have the heading Plan and Execute Life Cycle Management. And the first bullet point you mentioned, you will re-evaluate the appropriateness of the established preliminary acceptance limits. Is this for the first, for more than 100 batches? Was this agreed with the regulator agency that you will do this?
Yes, exactly. There was a post-approval commitment we had at the time of registration of the method. Of course, we have to think that the consistency limits basically take into account manufacturing variability and method variability at the same time. So it is, let's say, logic and correct to think that when more batches representative of the, of course, industrial manufacturing will be available, those let's say statistical ranges might of course change. So we are currently actually in our way of reviewing that, evaluating the need of reviewing those limits preliminarily established and during suitability.
Heidi, you're on mute.
Thank you very much for giving us this information. And I especially liked your last slide, slide number 9, where showed us what will be the next steps, the future steps, yeah? That you want to set up the antigenicity percent, which ELISAs. What is your perspective? How long will this take? Because this cannot be done in a short period of time. What is your assumption?
Well, let's say I cannot engage on timing. What we surely know is that it is a true change in paradigm, right? It's a true change in paradigm. We really have to be collaboratively convinced that this change is applicable, since we no longer measure efficacy in animals of the vaccines. We will consider that by the tools used, by the specific selection of antibodies that will be used, possibly, of course, have been strongly characterized for being seroneutralizing in vivo and in vitro of the toxins, that parameter will allow us to say, yes, I can have an antigenicity results on my batch according to an history of performance of those antigenicity results for the specific products, and use these as an element of proof of efficacy of the product. So it's a really change in paradigm, and we truly believe that it has to be built collaboratively with NCLs and all the health authorities and all the experts, also from other company that are in that perspective, that are moving in that perspective. And the back-to-back public-private initiative was very, very well aimed at this. It's a change in paradigm. We have to all agree on what is needed to move this step forward.
Thank you very much, Silvio.
Yes, there is just one awfully quick question in the chat. If it's possible to do a dermo test with one dilution, is it something that you have ever explored?
No. That was the main difference, if you like, with approach Pradeep presented earlier. So we wanted to move to the single dilution once we had proof by multimodal dilution that serology in guinea could be used. So we didn't proceed in that way. We didn't think to go to single dilution with, let's say, a clinical results, clinical outcome.
Thank you very much, Silvio, for your presentation and insights on the process that Sanofi is moving forward. Also to introduce the next session where we are actually going to get further information and insights about the possibility to transition to completely in vitro opportunities for tetanus, diphtheria, and their cellular pertussis. So it's my pleasure to introduce the first speaker of the session, which is Maxima Vermelin from Cenzano, again, same department from Morgana, so the quality of vaccines and blood products So thank you very much, Maxine, for being with us. Your presentation is on, and the floor is yours.
Thanks, Laura, for the introduction. Hello, everyone. It's a pleasure to be here to present our data today. And my presentation will talk about the development of a multiplex immunoassay that was developed at Cienzano during the VAC2VAC project to assess the antigenicity of diphtheria, tetanus, and pertussis antigens directly into the vaccines in view to replace the current warranty tests that are still performed on animal. So just a few words about Cienzano before to start. We are an official medicine control laboratory, so the main activity of our service is the batch release of biological medicinal products and especially vaccines through parallel testing and batch record reviewing. And these controls are independent controls and are performed before the marketing of the product. But in our service, we are also involved in some R&D projects. And in fact, the results that I will present today were obtained during the VAC2VAC project. That was a big project funded by IMI2 and EFPIA and coordinated by EVI. This project started in 2016 and ended in 2022. It involved a lot of different partners, more than 20 partners from different affiliations. And there were several vaccines manufacturers, different NCLs from several countries from Europe, academic lab, regulatory agencies, and also research organizations. And this is not represented on the slide, but EDQM, FDA, and Health Canada were also involved in the consortium. And the big objective of the project was to bring the proof of concept of the consistency approach for batch release testing of established vaccines using in vitro and analytical methods. So the principle of this consistency approach is that if you can demonstrate that your in vitro assay is well adapted to monitor key quality attributes of your product, such as antigen potency and antigen content, and you can demonstrate that these parameters are equivalent to those of batches that were already proved to be safe and efficacious at the clinical level, then you do not need to test new batches with animal testing because you can set acceptance criteria for these parameters that will ensure the quality control of the product. So the project was divided in several work packages depending of the method used. At Cienzano, we were involved in the second one, focusing on the development of immunoassay for the potency testing of antigen of DTwP vaccines. And we worked with two vaccines manufacturers and also with the team of Paul Stickings from MHRA in UK. So as you probably know, the potency assay for diphtheria and tetanus antigens are generally performed in mice or guinea pig by challenge assay, while pertussis potency assay is generally performed by serological assay. But in addition to ethical concerns that are associated with the use of animals for this testing, they are also expensive, they last longer, and they are more variable compared to in vitro alternatives. So for these reasons, during VAC2VAC, we worked with MHRA and the two manufacturers to develop immunoassay to directly measure antigenicity of antigens in the vaccines. So the team of Paul worked on development of ELISA for diphtheria and tetanus, and we worked on the development of a multiplex assay for diphtheria, tetanus, and pertussis antigens using the Luminex technology. So the principle of the multiplex using the Luminex is similar to those of a sandwich ELISA, but rather to be coated on an ELISA plate, the capture antibody are coupled to small beads, and each bead has a color code that can be recognized by the Luminex. So each bead is associated with the reading of one antigen. The two first steps consist to put the color-coded bead that are coupled to capture antibodies in contact with our target analytes. So in our case, the antigens of the vaccines. Then in the third step, detection antibody, which is coupled to biotin, is added and bind to another site of the antigen. And finally, at the fourth step, streptavidin conjugated to a reporter, the 5-core is added and binds to biotin. When the sample is analyzed by the Luminex, a first laser detects the color code associated with each bead, and the second laser determines the fluorescence intensity of the reporter. And as each color of bead is associated with one antigen, we obtained a measure of fluorescence intensity for each antigen for each point of dilution that was initially applied, and which allow to obtain a titration curve for each analytes. So in the end, we obtained a curve for the reference and for the test sample, and we can calculate the relative potency between the reference and the sample using a 4-parameter logistic regression. And we use Combistat software to do that. So if we compare it to classical immunoassays such as ELISA, the multiplex assay has the advantage to be faster, as it allows to assess all antigen in one run. If you observe the picture on the right, you will see that to test 5 antigens, ELISA will take around 30 hours, while it will take around 5 with the multiplex. But the inconvenience of the multiplex could be that the technology can be more difficult to transfer and is also market dependent. So during the testing made in parallel with the team of Paul, we both used the same pairs of antibody for diphtheria and tetanus. And these antibodies were monoclonal antibodies that were in depth characterized during the project to be sure that they are stability indicating. MHRA worked on the development of the ELISA, and we did not observe major differences in terms of results and variability with the multiplex for diphtheria and tetanus. So yes, during this work, we made experiment on 8 different DTwP vaccines from 2 different manufacturers, but the assay was not evaluated on wholesale pertussis vaccine. Now I will give you some data from experiments performed during the development of the assay. So we tested the specificity of the assay using dropout samples obtained from the 2 vaccine manufacturers. And in fact, each dropout corresponds to the complete formulation, but lacking one antigen. So for example, the DT dropout is the complete formulation without the DT antigen, etc. for whole dropout. And we observed a good specificity in the assay for whole dropout, as the signal for the lacking antigen was null in all cases, except for the TT antigen of the manufacturer A, for which a low signal was observed. And in fact, it can be explained by the presence of TT that is used as a conjugate for the antigen of flu in this formulation. But otherwise, we did not. We observed a good assay specificity. We also evaluated the cross-reactivity by comparison of the signals obtained for each antigen in the single and in the 5-plex format. The results showed very good correlation between mono and multiplex assay, suggesting that there is no major interferences in the assay when used in multiplex. Then we just evaluated the assay by titrating non-adsorbed and adsorbed antigens from manufacturer A and B, and we observed that we can obtain those response curves with both adsorbed and non-adsorbed antigens from both manufacturers, which was a good starting point. And we also obtained good dose response curves with non-adsorbed and adsorbed standards or references obtained from MHRA. Finally, we also evaluated the technical variability of the assay by doing a transfer study with one of the manufacturer. The data presented here are for two different products from manufacturer B. So a high-dose DTwP vaccine and a low-dose DTwP vaccine, so a booster vaccine. And for each product, we took two batches. One batch was assigned as the reference, and the second was assigned as the test sample. And these batches were titrated using multiplex assay in duplicate 3 times and in 2 different labs. And then the relative potencies of the test sample were calculated compared to the reference using the Combistat software. And the results of relative potency for each antigen were used to determine the geometric coefficient of variation. And as you can see on the right column of the table, these coefficients were below 10% in all cases, suggesting a good level of precision of the assay and a low technical variability between the two labs. So now we can look at the potential application of the assay. So a question that we can ask is what a good immunoassay should detect in the frame of quality control of vaccine. And to answer this question, we refer to the guideline of European Pharmacopoeia in chapter 5 to 14. And what this guideline says is that the assay must be able to assess K quality attributes in the product. And as a first K quality attribute, we try to see if the assay was able to assess antigen functionality in the product. And to evaluate this parameter, we generated voluntary altered vaccines by heating vaccines and at different temperatures, or by mixing the vaccine with hydrogen peroxide. So here you can observe the result of the titration of heat-altered vaccines for the 5 antigens. In this case, this is a DTwP product with diphtheria and tetanus toxoid and 3 antigens of pertussis. You can see on 2 all graphs that compared to the reference blue curve, which represent the non-heated sample, that there is a shift of the other curves compared to this reference, indicating that the assay detects the antigenicity modification or alteration. For diphtheria, tetanus, FHA, and protactin, the signal decreased compared to the reference, which seems quite logical, while it increased for the pertussis toxoid. After discussion with the manufacturer, it appears that this process can be due to epitope release on the pertussis toxoid induced by the degradation due to the breaking of molecular bonds created during the detoxification process. And we observed this phenomenon for PT for one of the manufacturer, but not for the other. So this is it seems that this is product specific. On this second slide, you can observe the same kind of result, but this time following an alteration of the product with hydrogen peroxide. And again, we observed a shift of the curves following alteration of the product with different concentrations of hydrogen peroxide. So with this assay, with this test, we confirm that the assay is capable to assess antigen functionality in different DTwP vaccines. Has the assay detected antigen degradation induced by heating and hydrogen peroxide? Then we tried to demonstrate that the assay can also assess a second important quality attribute, that is the antigen content. So we generated under and overdosed formulations of the vaccines, and we titrated these vaccines with the multiplex assay. So here you have a first example for an under-formulated DTwP vaccine. We prepared formulations containing 0, 25, 50, or 75% of antigen, and we compared it to the control complete formulation that was fixed as 100%. And if we look to the recovery calculated between theoretical and calculated antigen relative amount, we observe that there is a good recovery between 80 and 120%. So we can detect if there is an abnormally low amount of antigen in the formulation. Here, this is a second example for another under-formulated product containing also pertactin as additional pertussis antigen. And again, we observed a shift of the curve when we decrease the antigen concentration in the product, indicating that the assay can detect the abnormally low amount of antigen in the formulation. Finally, a last example, but this time we made the opposite exercise. We generated overdose formulation of 150 and 200% of antigen compared to the normal formulation. And we could detect the abnormally high concentration for all 5 antigens with an acceptable recovery percentage. So this indicates that the assay can also detect abnormally high amount of antigen in the formulation. So with this second set of experiments, we demonstrated the capacity of the assay to detect abnormal amount of antigen in vaccines, which is an important parameter for the quality control of the product. And finally, we tried to see if we can monitor the consistency of the potency results with the immunoassay. So here you have an example of results that could be obtained for the batch-to-batch consistency monitoring with data for DTwP vaccines from the manufacturer Hay. And the idea of this approach is to control that the production is consistent and that the quality of the product is maintained between the different batches compared to a control batch. Ideally, the control batch should be a batch that was already proved as safe and efficacious during clinical testing. And here, this is an example for only 12 batches, but we can see that thanks to good manufacturing practice, the variability of the antigen potency between batches is low if you look at the upper and lower limit of the 95% confidence interval in the table. And in fact, if you look at the two columns on the right of the table, at the lower and upper limits expressed as the percentage of the relative potency, you can see that these are around 80 and 110%, which suggests an acceptable variability from batch to batch. So this was just an example of the application of the assay to monitor the batch-to-batch consistency. Yes, so these results and results obtained for the ELISA by MHRA aroused a great enthusiasm from members of the consortium. And in fact, one of the manufacturers decided to transfer the ELISA method. And the second one to transfer the multiplex assay. And according to what we know, they started to work on it internally. To our knowledge, discussion with regulators has also started to obtain guidance on how to proceed to implement the in vitro test as a replacement of in vivo potency tests. So this is a good point. And another important point is that all the antibodies that were used for the development of the assay are now available, and you can buy it on the website of the MHRA. And you can see the name of the different antibodies in the tables of on the middle of the slides. In this table, you can also see that there are anti-FIM antibodies. They are also available, but these antibodies were characterized during VAC2VAC, but they were not used in this study as vaccines that were tested did not contain these antigens. Also, the methods that were developed for the multiplex testing and for the diphtheria and tetanus ELISA were published in open access, and they are fully available on the internet. So you can you have a full access to it. And yeah, now it will maybe take some time to implement the assays in routine, but the important thing is that a mindset operated in the head of the regulators and manufacturers during the VAC2VAC project, and everybody seemed to be convinced that this is possible to move towards animal-free assay for the quality testing of vaccines. Thank you.
Thank you very much, Maxime. We have a good number of questions in the chat. I think that some of them can be answered by you, but also by Paula. So I think that's we can probably have a little bit of a discussion here in case Paul or others would like to jump in. The first one, I think that it's so from Alexandrine Maes from WHO, if there is a scientific reason why it was not tested on the whole cell pertussis vaccine, and if there is a possibility that the multiplex assay would be used also for the whole cell. This is an important point, and hopefully we will have also a little bit of a discussion in the next session. But please, if you would like to argument about this.
I have no explanation of why it was not tested, because I think, as Catherine said yesterday, the consortium was in Europe, and we mainly use the vaccine that are used in Europe. And in Europe, we don't use the whole cell pertussis vaccine. So, but I don't know what it could give as results if we tested one. I think, Paul, you tested some vaccine, but only for the DNT. So
that's right.
I don't know if you observed difference with AP or between AP and whole cell pertussis.
No, I mean, very limited testing, but only for the DNT components, as you said, and it looked promising, as if it might be applicable, but applying it to the whole cell component is a completely different challenge, and I think we might hear more about that at the end of towards the end of the webinar.
Thank you very much, Maxime and Paul. There is another question about whether the vaccines that you tested were adjuvanted or not, if the adjuvants give any kind of interference, or you experience any interference issue while using your methodology.
During the project, it was decided to not investigate in depth the desorption step, because if we want to apply the immunoassay, the desorption can change the integrity of the antigen. So we are not sure if the desorption step will alter the antigen or not. But I think the idea is the assay could be applied as they are, and maybe had a test for the completeness of adsorption, for example, or of the final product.
Yeah, and just to say, we have some limited data on desorption, and I'll show some of that in a few minutes.
Thank you, Paul. Then moving ahead, it's a clarification about the term functionality, Maxime, that you used in your slide where you were saying that what kind of parameters you actually worked to double check based on the chapter 5.2.14. So if you can clarify the terminology that you used, the functionality of the antibodies.
The antibody were characterized before, earlier during the VAC2VAC project, and different tests were performed. This was their ability to bind the native and the adsorbed toxins, and also the altered antigen, especially by heating the antigen, just to see if which antibodies can detect the better the alteration of the antigens. And these results are published for diphtheria and tetanus, and we want to publish also for the pertussis analysis of antibodies, but this is underway.
Thank you, Maxime. And this is actually, I think that answer to the other question about whether you map the protective epitopes of diphtheria and tetanus toxoid, or if there is any plan to establish it.
I mean, do you want me to take that one, Maxime? I think it wasn't done during the project. We focused on looking at functional relevance. So, for example, do the antibodies neutralize toxin, which we showed that they do in the case of D&T, and I think at least one, if not both, of the pertussis toxoid antibodies as well would prevent CHO cell clustering, for example, and also relevance in terms of the ability to detect quality changes after exposure to heat, for example. But I think it's an interesting point, and it's potentially something that might need to be explored further as assays become formally validated.
Thank you, Paul. 1 second, because there are a lot of questions coming in at the same time. I think that's there are a couple of questions that I think that we can take after your presentation, Paul, that it's about the next steps in terms of whether to establish in vitro acceptance limits or assess the discriminatory power of in vivo versus in vivo. And then if there is the opportunity to do, or if there is any plan to do any collaborative study on this. I guess that's those comments, we can take it after your presentation, Paul, if you don't mind. Let me see if there is something else. Okay, so I think that's the other, we can take it after your presentation, Paul, so then we can proceed. So I think that's, Ademarie will introduce you.
I would like to ask Maxime 2 short questions. The Luminex is, I think, a costly equipment. What do you think about this? Because said one manufacturer will set up the ELISA, and the other one will also implement the multiplex assay. So I never made a research, but how costly is the equipment?
To be honest, I'm not the person who bought the equipment, but I think it's around 60,000 euro.
60,000 euros.
I think.
And as you said, the 2 manufacturers, the one will set up the ELISA and the other one the multiplex. Will you then, as an OMCL, the releasing OMCL, also then set up for the first manufacturer the ELISA method, or will you stay with the multiplex?
This is not already clear how it will be organized in the department, because we do batch release for these 2 manufacturers. So as the methods seems to be product specific, probably the idea would be to use the 2 methods, but it's not already clear. We have to talk about it internally.
Yes, this I understand completely, because if there's an initial OS, how shall you argument this then if you use different methods, for example? But it will be your decision. Okay, thank you very much for this, Maxime.
Thank you.
And with this now, Paul, you're already on the screen. So with this, I introduce you now to hold your talk. Paul Stickings, he was part of the work package 2, especially in the DTwP project. And in this work package, you did created lots of ELISA data on this vaccine. So I'm very happy to listen to your talk.
Okay, thanks, Heidi. You can see the slides, and are they moving?
Yes, they're moving. Thank you.
Okay, thanks. So a lot of what I'll talk about has very similar themes, of course, to what Maxime has spoken about, but I think I'll bring in a bit more detail for some of the considerations for taking alternative assays forward. And the focus that we have at the moment are on the products, particularly the legacy vaccines, where in vivo potency assays are still used routinely as part of the control strategy post-licensure, and obviously diphtheria, tetanus, pertussis are among the best examples of that. And I think we acknowledge increasingly that in many, maybe most cases, they're not necessarily the best tool available to us, the best assays that can be used for the purposes of routinely monitoring consistency of quality and consistency of production, to give the assurance that new batches are consistent with those shown to be safe and effective in clinical studies or through routine clinical use. And one of the main reasons why they're not necessarily the best tool for the job is because they have inherently high variability. And the data that I share with permission from Emmanuel Coppens from Sanofi that she first presented a few years ago at a meeting is quite striking in that it shows that for a tetanus vaccine and for a diphtheria vaccine, you have to make and deliberately make very large changes to the amount of active substance in those vaccines before they no longer meet the requirement, the minimum requirement for potency in the in vivo models. And that's due, at least in part, to variability of those models. And then we've had some nice publications recently that attempt to quantify just how variable some of these models are for D and T and for some other vaccine components as well. And Maxime has already really nicely introduced the VAC2VAC project, so I won't repeat that. But our focus in that project was on immunoassays and specifically ELISAs that potentially could substitute for the current in vivo potency assays for the routine control of the critical quality attribute of potency in diphtheria and tetanus vaccines. And the methods that we developed have been published, and they are essentially the same method, with the key difference being the antibodies that are used to capture and then detect the antigen. And all of the data, I'm not going to go through the data, but what we show in those publications is that the methods are widely applicable to different products, including for the veterinary community and the audience, veterinary tetanus vaccines. We show that they are specific, sensitive, very precise, using assays and studies very similar to what Maxime showed just now. Some good evidence that the assays are likely to be stability indicating, and also successful transfer of the methods to other laboratories. So this took us as far as a good demonstration of proof of concept. It wasn't within the scope of that project to validate the assays, because that is always going to be done by product manufacturers. And as Maxime alluded to, that work is ongoing as we speak in some companies. So I'm going to focus now in the last part of the talk on some of the things that need to be considered as these methods are validated and hopefully implemented. And they include the availability of reagents, the choice of a reference vaccine or reference antigen, and then something about desorption. So we'll take them one by one, starting with the reagents. And one of the barriers to implementing alternative assays that have been developed is often the availability of critical reagents that you need to perform that assay. And I think one of the major successes of the work package that we were involved with in VAC2VAC is that we got the agreement and the cooperation from the owners of the critical reagents that we use in the methods. So the method that Maxime described and the methods that I've talked about. They all agree that these antibodies can be shared for other organizations who want to further develop and hopefully implement these methods. So for all of the antibody pairs, for each antigen, purified MABs are available through the catalogue that I think many people will be familiar with, people that, for example, already use WHO standards. They're available from the same place, from the NIBSC catalogue. And I thank again all of the organizations who cooperated to provide that agreement that they could be used for this purpose. And just to say Maxime referred to the publications where we summarize the characterization of those antibodies. And for D&T, I've put the links at the bottom. So these slides will be available after the webinar for people to further explore that information. So next, let's have a look at the choice of a reference vaccine or antigen. The assays, the ELISAs, provide a measure of relative antigen content and quality. And you need to have a reference vaccine or a reference antigen in the assay, which of course needs to be stable, it needs to be qualified, and it will need to fulfill validity criteria for linearity and parallelism, as you would expect for any quantitative assay of this type. And for diphtheria and tetanus, there's actually quite a few different options that are potentially going to be suitable. We have WHO standards, we have adjuvanted standards, and we have non-adjuvanted for D&T. And then the manufacturer, of course, has drug substance, adjuvanted and non-adjuvanted in some cases, and they have drug product. They have the same product that is under test, but potentially we could think about a product from the same company serving as a reference for other products from the same company. And so we've looked at all of these options within the project for a selection of different human vaccines. So I'll talk you through what we observed. We had 7 different products, and we ran the vaccine in the ELISA, comparing the dose response to possible reference vaccines or reference antigens. And here, this is data for the WHO international standard and the manufacturer drug substance. In both cases, non-adjuvanted. And this is data for the diphtheria antigen using the diphtheria ELISA. Crosses indicate where the dose response was not comparable between the reference and between the product. Where we have a tick, that indicates that the dose response was comparable. And where we have a cross and a tick, that means the conclusions are equivocal. So we looked at 2 batches in each case for all of these 7 products. And where we have a cross and a tick, it means that for one batch it looked okay, for the other it didn't. So an equivocal result. But generally, looking at that picture, I would conclude that non-adjuvanted material may not be a suitable choice as a reference for the diphtheria antigen, at least in those products that we've evaluated. It's a different picture when we look at adjuvanted reference materials. So we have, again, the WHO standard, and then we have manufacturer materials, drug substance, and drug product, either exactly the same drug product in this line or a similar drug product, but coming from the same manufacturer in the bottom line. And across the board there, we saw really nice comparable dose responses. For tetanus, picture was broadly similar, perhaps looked slightly better in terms of the non-adjuvanted the non-adjuvanted reference options. And for adjuvanted material, with a couple of exceptions, again, it looked pretty good across the board. And taking all of that together, I think it's clear that for a number of products, there will be more than one option that potentially would be suitable. And then the choice will come down to other factors, logistical factors, thinking about availability of materials, how stable they are, and if it's something that doesn't have long-term stability, the implications for managing that program over time, having to replace and then managing the potential for drift, all of those things will come into consideration as they would for the management of any reference standard program. But I think it's encouraging to know that there are potentially multiple options that are going to work. Lastly, I'll focus on desorption, which this came up in one of the questions. So all of the human vaccines, at least, contain aluminium adjuvant, and all of the vaccines that we looked at in the project contained such adjuvants. Some of the tetanus vaccines, the veterinary tetanus vaccines, don't have an aluminium adjuvant. Some of them do. And what we know about absorption and, antigen and epitope accessibility to an antibody is that the picture is likely to be different depending on the product and depending on the antigen, because the amount of adsorption and whether that adsorption affects epitope availability is not something you can predict. It's something you have to evaluate. And it is something that we evaluated to a limited extent with a small selection of products. And I'll talk you through how we did it and then what we observed. So the first thing we can do with a vaccine sample is run it in the ELISA without any kind of pre-treatment of the sample. And that provides us a measure of the detectable antigen in that vaccine. We can spin the vaccine to pellet the adjuvant, and any antigen absorbed will obviously stay in the pellet, and any non-adsorbed antigen will remain in solution. So we can test that supernatant, and that provides a measure of the non-adsorbed antigen in the vaccine sample. And for some vaccines, when we do this, we see nothing in the supernatant, which tells us that in that product, the antigen is completely absorbed. And that's quite normal with some products and some aluminium adjuvants. We then need to understand the total amount of antigen in the vaccine. And to do that, we have to apply a desorption step. And there are different ways of doing this. I'm just showing one example of using sodium citrate to dissolve or remove the antigen from the adjuvant. And so once that step is complete and you centrifuge the sample, all of the antigen will have been desorbed and it will remain in solution. The adjuvant will be pelleted, and you can then get a measure of the total antigen that's in that sample. And once we have these three measurements, we can firstly work out the percentage of the antigen that is absorbed. And importantly, we can understand, based on the difference between these two measurements, the proportion of the total antigen that is detectable in the vaccine using the ELISA when we don't do any pre-treatment of the vaccine at all. So as I said, it's something that you have to evaluate, and it will have to be done during validation, and it will have to be done for every product, because you will see different outcomes depending on the product. And I'm going to just pick one example for one product and looking at both antigens here. So here we have data for a tetanus antigen in a combo vaccine from one company. The antigen is 100% absorbed. It's completely absorbed to the adjuvant. But once we go through the process that I described on the previous slide, we can conclude that when we run the vaccine sample without any pre-treatment, we are detecting all of the antigen that's in that vaccine. However, in the same product for diphtheria, again, the antigen is 100% absorbed, it's completely absorbed, but having gone through the desorption process that I described and then comparing the measurements, we can conclude that we're only detecting about 2/3 of the antigen in that vaccine when we don't make any any desorption of the product, and we just run the sample in the ELISA. And then that brings into focus, a really important question, because if you can demonstrate that you are detecting, all of the antigen, as I showed with the tetanus example, or maybe nearly all of the antigen, then it seems fairly clear to me that you wouldn't need to routinely apply a desorption step. Once you've demonstrated that to the satisfaction of the NRA, of course. But for some products, it's clear that you would not be detecting all of the antigen without any kind of pre-treatment. You will be detecting a proportion. In that example I showed, it was 63%. In other products, it was as low as, I think maybe as low as 30 or 40% in some cases. And there will be a threshold at which you would conclude that you do need to routinely apply a desorption step. I think identifying what that threshold is really quite difficult. I mean, I really don't have a view on what it should be, but it's ultimately a question that has to be addressed and looked at in validation and then in discussion with the NRA. And then Maxime, the other important factor, Maxime touched on this, is that if a desorption step is to be applied, it's important to understand whether the process itself impacts the integrity of the antigen, and also whether the process of going through a desorption step impacts the performance of the assay, for example, the variability of the assay. And we've looked at this admittedly to a limited extent, but what we see is quite encouraging. Adding a desorption step, it increases the assay time by a day. So it does have that impact, but we didn't see a significant impact on integrity of the antigen, and we didn't see a significant impact on variability of the assay. And then just to finish, a couple of other considerations. So one is to do with the reference standard. I mentioned that the use of WHO standards look like they will be an option in some cases, but I think it's clear that they would be used differently. The data coming from the assays would be interpreted differently. And I think one of the consequences is that comparing potency of different products through laboratory testing will not really be possible following a switch to in vitro assays like this. And as others have mentioned, acceptance criteria, so specifications, are likely to be based on consistency and therefore product specific. And where these methods, whether it's ELISA or Luminex, are taken up by manufacturers, I think it's highly likely that they will make certain modifications as part of the validation process. They could be minor modifications, or they could be quite significant ones, for example, using different antibodies in some cases. And some products will require desorption on a routine basis, others may not. So there'll be lots of different assays out there, I think, in the future, hopefully. And that poses a challenge for NCLs. Heidi, this comes to the point you mentioned, and it's something that NCLs would have to think about where independent testing is done. Do they adopt the different methods that are being used in different manufacturers for testing those different products, or do they try perhaps and adopt some kind of universal protocol, provided it's shown that it does work for the different products? All of these things are important considerations for the future. So I think, the work that Maxime described, that I've described, has a lot of promise, would bring significant advantages, I think, in terms of having improved discriminative power for monitoring batch quality, much faster compared to current in vivo assays, and of course removing the need for animals completely from the routine control strategy for potency, at least. And there are definitely challenges still to address, but I think the experience from other efforts and the expertise that we have across the different stakeholders means that those challenges can hopefully be met. And I'd like to acknowledge, as Maxime did, all of the people who have been involved, particularly from that VAC2VAC consortium, and I'll finish there. Thank you. Happy to take questions.
Thank you very much, Paul, for this really fantastic presentation where you showed us lots of data. As you just said, what shall we do if a manufacturer does another method and the OMCL, the NCL? the most important thing is that you have an agreement on the specifications, because if you use another method as an OEM cell manufacturer, you will get different data. And this there should be a letter, an agreement letter, something like this, that an OMCL is then allowed to say, hey, might be an OS. And we did this, and this works. So just take the manufacturer, stress him on this, and it should come to an agreement. This is just my own experience. We got some questions here. Laura, how shall we proceed? Yeah.
So I think that there we can probably bring up and please Silvio jump in because you can take the floor. You made some interesting point about the next steps. Similar, there is again a couple of other questions wondering about. Okay, so what is going to happen? There will be an international collaborative study, or also the comment from Sajan about the uptake of this methodology inside the pharmacopoeia, for example. So I would actually like to to have maybe Silvio, Sajan, if they really want to comment their questions, so then we can have a little bit of a discussion about this since the topic is super important.
So I mean, I can answer the question about collaborative study, maybe take that one first. So I'm not aware of any definitive plans for such a study. But I think Catherine's online. She could maybe speak to EDQM. I know it's something that's under consideration, but not with any definitive plans. I mean, from my perspective, I think we'd be very happy to be involved and support such a study if there was something done in the future. I see Catherine is.
Did you want to comment?
Yeah, please, Catherine. Thank you.
Thanks, Paul. I think your summary is pretty good. So, I mean, the EDQM has been involved in the VAC2VAC project from the beginning, and there's always been a discussion of the possibility to have some sort of collaborative study to try and help with the implementation of these assays. But I think you've seen, and we've seen together, between the presentation that Max gave and your presentation, that there's quite a lot of differences in the types of approaches that might be taken. And we have the multiplex, we have the simplex, we have lots of different kinds of vaccines. So the design of a collaborative study would be quite complex. And I think the real goal of a collaborative study should have some added value to the situation in general. We've seen the different manufacturers who are invested in the method already are taking, in some ways, their own paths and adapting some elements with respect to the antibodies that are being used. So I think the idea of having a study to help with the implementation is still open, but I think we need to really have a good idea of what the added value of that study would be, and hopefully it would help to promote implementation on a wider scale. But I think we have to be realistic. It's not going to be a product by product validation of the use of method A or method B or whatever method with a key in hand recipe for each company, each manufacturer, each NCL. I think that there's going to be some discussion needed as to how to go forward in that context. But there is, let's say, an interest in the potential for such a study, but the actual context has to be evaluated, and it would only go forward if there was a real added value.
Thank you, Catherine. And taking up, since you are sharing what is the DQM perspective, there is any plan to introduce any kind of language in the European Pharmacopoeia that is going to allow this methodology to be taken up if, as we have seen, probably some companies are already working on implementing those methodologies within their products?
Yeah, no, I think that's under discussion in the expert groups. So it would be group 15 and group 15V, and the experts there are kind of trying to see how they can address that, because I think it's a little bit of an intermediate stage. We know that these possibilities exist. We know that they could work. Right now, we haven't got anything at an approval state for any registered vaccine. And, you know, the Pharmacopoeia works with methods that have been approved by regulatory authorities, or things that have come out of our collaborative studies. That doesn't mean that there may not be possibilities for what people like to call door openers, but I think that's all within discussion at the level of the experts right now. So I think there may be some possibilities, but it's up to their discussion.
You, Catherine. Then getting to the question from Silvio about what are the next steps. So within you, Paul and Maxime, are you working to establish in vitro acceptance limits, the discriminatory power of those assays, or is there still some work ongoing in further develop those methodologies?
So I think there is, but not by us. I mean, our work effectively stopped with the Vac2Vac project, you know, when that ended. But yeah, these are precisely the things that manufacturers will be looking at as part of further development and validation studies.
And maybe I just add the caveat that I forgot to add, but is always true in the European Pharmacopoeia. The general notices and introduction always allow the possibility to have a different assay than what is indicated currently in a monograph if you can show that it is a suitable alternative with the data that you have. So even now, there's no block, but I understand that especially the manufacturers are more comfortable if there's more concrete language in the Pharmacopoeia to give them some reassurance.
Thank you, Catherine. Very important remark. So no one is actually stopping, at least within the European context, to go ahead. I have one other interesting general question. Then we can go to the ones that are very technical one. It's about whether those methodology can also be used for the development of new vaccines, or they are actually very focused only to target the efficacy once you have the human clinical studies that are done. So is this something that's it's an approach that could be considered while you develop new products?
Well, yeah, I mean, if we're talking about new DTwP products, then yeah, I think it is.
And
I
think,
mean, sorry, go on, Catherine.
No, you're probably going to go a few more.
Yeah, no, go on, please.
No, I was going to say, and I think it's absolutely critical to use these assays during the clinical development stage to have information with the assays that you'll be using in vitro on the actual products that are going into clinic to see how the values you're seeing in the clinic relate to the values you're getting from your potency tests in vitro.
Maybe a reflection from the manufacturer perspective, definitely talking about consistency approach. It's much easier to see this change from in vivo to in vitro for well-established products that have, let's say, a long proven history of clinical efficacy. And is that, you know, that question on how to set acceptance limits, how to demonstrate clinical relevance of those acceptance limits is definitely easier, let's say, to produce that evidence with well-established products for which we have multiple years of data and also clinical efficacy. But that is the very core subject we of course we are working on. We started to discuss with authorities how to possibly show that by this consistency approach we are in control of any eventual change, for example, over time of the antigenicity profile, whereas no change in previous in vivo followed quality attributes will not be possible to be seen. So there are definitely many questions to be answered. And again, we are really convinced that that has to be constructed together with health authorities and NCL to have really convincing significance of this change in paradigm in acceptance limits.
Thank you, Silvio. Indeed, thank you for your perspective. So, do you? There are really a lot of questions. I don't know. Do you have any preference from where to start? I think that we covered a little bit the impact about the desorption step via Paul's talk. I don't know if maybe you want to get a little bit more further into the details. Silvio again was asking about availability, the available data relative to the impact of the desorption on the antigen integrity itself, and or to the matrix alteration in the combination of vaccines. I don't know if you had further data on this.
I mean, it's not, I'd have to check. I don't think it's in the published papers. But essentially what we did is we took a non-adjuvanted drug substance, a toxoid, and put it through the desorption step and compared before and after. And we saw no significant difference in terms of the antigenicity measurements that we were doing. But, we only did that with probably 1 or 2 toxoid samples. And again, it's something that needs to be done, for every product, for every toxoid that's produced. I didn't get the second part of the question.
Eventual matrix asset. I think you perfectly answered, Paul, I guess, okay, to the question, meaning how do we assess if, let's assume that we can gather antigenicity results, consistent antigen results with both a desorption step or without a desorption step, not actually only in adsorbed DS, but in very complex hexavalent matrices, for example. So I think one question we are trying to answer to, but it's definitely one question from the manufacturer, is that should we dissolve and associate these antigenicity results as representative of our complex matrix combined products, or should we stay with an antigenicity approach without desorption, and therefore almost by definition representative of the product itself because it's untreated before antigenicity testing. So these are basically the type of questions, of course, I think we are asking ourselves, and I think also other companies are currently trying to address.
Yeah, and it's not a straightforward, unfortunately. I think it really comes down to, in the combo, what proportion of the antigen are you seeing with whatever assay is being applied? And if it isn't 100%, how far away is it? And also, to what extent can you be confident it's representative of all of the antigen in the combo in the vaccine, so including the antigen that's absorbed? And I think that can only be determined by going through those desorption studies. So I think at least in development stage, those desorption studies have to be done. And hopefully the picture becomes clearer after that. But it is a complex aspect of these type of assays, I think.
Thank you, fully agree.
I have an additional question here. Do you have a map of the protective epitopes of DIP and TET toxoids? Is there any plan to establish it?
I mean, no plan from our side. And again, within the VAC2VAC project, there was only scope to do so much. I think it would have been nice to do a bit more work in terms of the characterization. So we have no plans, but I can recognize that it is something that I think would potentially add value and add confidence to, taking these assays on further. So I think some of the protective epitopes are known, but the mapping, at least for D&T, hasn't been done for these maps. But what we do know is that there is, they are functionally relevant in the sense that they neutralize toxin.
Yeah, thank you, Paul.
I mean, am I allowed to answer just one that I've spotted, which I think it's an interesting question. It's about the measurement of LF. And of course, that's a measurement that's already done. But I think it's important to point out that although the assays that we've talked about today, Maxime and myself, they are quantitative, but the measurement is relative. So it's not an absolute measure of antigen. And I don't think, although we haven't formally studied and evaluated a comparison to, for example, a flocculation test, I'm pretty sure that they would not compare. But that doesn't matter because that's not the purpose. The purpose is not to get an absolute quantification of the amount of antigen. It's to measure consistency. It's to have a relative measure that you can then use to look at consistency. And of course, we need to be sure that the assay, the antibodies are able to spot inconsistency. So if there is a quality defect, a consistency problem, we need to know that the assay is going to see that. And I think based on the work that we've done and we've published, we have some level of assurance that we will. And then further studies will of course be done within the manufacturer development and validation to provide, I think, additional assurance that they will.
Paul, that's actually very interesting because there are additional comments in this chat is actually questioning whether the LF is probably more relevant than the in vitro tool because of the potential parallel of the data with the clinical trials.
Well, I mean, LF, so the LF is an amount, gives you the measure of an amount of antigen, but there's no correlation between amount of antigen and potency because, in vivo potency, because that depends on so many other factors, other components in the combination, adjuvant, degree of absorption, all of those things will impact what you measure in potency, and there is no correlation with the LF value.
And it comes down to the definition about the potency. So exactly what does it mean doing a potency test, I guess, here, right?
Yeah, I mean, it's a measure. It is a measure of the ability of the vaccine to elicit a functional response or an antibody response of some kind, but it simply doesn't correlate to the amount of antigen in the vaccine because there are too many other factors that influence the in vivo part.
Thank you, Paul. I think that we really had a very interesting discussion. If you agree, we will probably proceed to the whole pertussis piece, and then we will still have a little bit of time to get into the details of these different methodologies and understand their concrete implementation or what we still need to put in place. So thank you, Paul, and thank you, Maxime, and thank you all for all the questions that you raise. Hopefully we were actually able to provide you some interesting insights. So since some of the question that you had so far in the chat was also inquiring about the possibility to use some different opportunities also for the whole cell pertussis, I'm actually going to give you some ideas about that. So again, within the project that we are running, we are getting in touch with a lot of stakeholders, being both regulators and manufacturers that are producing and releasing the whole cell pertussis. And the question about I'm actually going straight to the point. The question about how to prioritize the in vitro opportunities for both.
Laura,
yes.
Laura,
Can you hear me?
Sunil wanted to start.
Oh yes, sorry. I just stop it. Yep.
Yeah.
Sorry, Sunil. I give you the floor immediately.
You may continue, please, Laura.
No. Are you sure?
Check my video and mic
okay.
whether it works. Yeah.
You can jump on it afterwards. Yes, thank you. Thank you very much, Sunil. Okay, so give me a second that I go back. Sunil will get into some of the complexity of what I'm going to say right now, just shortly after these parentheses about the whole cell pertussis. So we have been seeing that discussing with all these different stakeholders came out with very important information about how animal testing is still a major issue to tackle for the tetanus diphtheria cellular and whole cell pertussis. So the amount of animals that are used, especially for the potency, it's very important. It's very relevant because sometimes you start from a minimum of 140 animals per batch for the in vivo challenge, and considering that hundreds of batches are producing annually, the impact in terms of animal use is pretty important. Also in terms of the timelines that are needed in order to release a batch. What it came out is that it's still very important to follow the conversation and support all the discussion to prioritize the transition from in vivo to in vitro for those type of products, because the reliance on animals are still requiring to have a very well-trained personnel. And actually, we saw also from the initial conversation from the single dilution that personnel might be trained. You need to maintain the knowledge on how to perform those methodology, and many of those in vivo methodologies really you need to make sure that the personnel really adhere to the safety procedure, because they are handling with toxins and bacteria, and sometimes there is the risk to be infected. So there is really an important knowledge that needs to be maintained while you keep the in vivo testing into your pipeline for the release. So the investment on transitioning from in vivo to in vitro requires that there is an increased investment on the different methodology to secure safety and potency testing. You need to make sure that the critical reagents are available, and you heard from Paul and Maxime about how important it is and how that was part of the VAC2VAC project. So availability, affordability, and accessibility of this material is important. The cost of the equipment is also something that needs to be considered. And at the same time, also understanding what does it mean to transfer those methodology from a company setting to the national control laboratories, and making sure that the knowledge transfer and the method transfer goes flawless. And last but not least, there will be also very important to make sure that the regulations are going to get updated and allowing the investment of this company. The whole cell pertussis, you are probably very familiar with that, but the only official way to release the whole cell pertussis for the potency testing is the Kendrick test. It was actually developed a lot of years ago. So the first publication is from 1947, and it's a intracellular challenge with a specific strain of the Bordetella pertussis. And this is again requires a lot of time because, of course, there is an immunization passage, flu, and then the challenge and the analysis of the results. So, what is the impact on the different group of mice? And there was a lot of work in trying to find an alternative to the Kendrick test. And again, you can see here in the 3 boxes where there are 3 main opportunities that have been explored. And one is a respiratory challenge, and another one is the nitric oxide production by the macrophage for mice immunized with the whole cell pertussis in response to the in vitro stimulation with bacteria antigens. The first 2 that have been tried, they were not further explored because they are not QC ready. So there was no further investment on these things, while on the serology approach was something that, as you can see, has been actually interested by a lot of stakeholders that worked for the last 30 years in exploring how this opportunity could be So you can see here in one box all the studies that have been started since the 1994 till the project that was led by DC Women to assess the feasibility of the serological potency assay for the whole cell pertussis contained vaccine. The conclusion of that study is going to be hopefully soon published into a WHO publication. But again, the results of it was that there is the need to further optimize the ELISA and also to further optimize how the in vivo piece of the serology is going to be done, because the results were a little bit variable between the different laboratories that participated within this project. We had 10 different laboratories, national control laboratories, and companies producing the whole cell pertussis. So this is something that could be hopefully still considered or can be discussed whether there could be an opportunity to further explore. And again, and it comes to this very short list of considerations where, again, when discussing and thinking about the future of the testing for the whole cell pertussis, what it should be, the future of it. We got a lot of data about the serology, although, as I say, there are still some complexity and a lot of consideration that needs to be taken on the difficulties to implement it that could be linked to the product specificity, to the use of international versus in-house standard. They need to better optimize the dilution that needs to be performed for the serology. So the question here is that, what is the future of it? Shall we continue to discuss and maybe further invest on the serology, or again consider something very similar to what the VAC2VAC project put in place for tetanus, diphtheria, and cellular pertussis, knowing that the whole cell pertussis would require a completely need to do the characterization of the bacterial itself, because what are the critical quality attributes might still need to be identified. And this is something that is not going to take a few years. So we have been seeing that the VAC2VAC project required a lot of years to come to the results that Maxime and Paul just shared to you. So again, we are here hopefully today also to understand a little bit how the future of the whole cell pertussis might look like. And without this, I cannot give you an answer. The answer is probably like in a further discussion and understanding exactly about the possibilities that we all have within the different manufacturers and the regulatory authorities. But there is an interesting work that has been done recently by our next speaker, and I hope that Nick can bring up the work that he has done within the whole cell pertussis bacteria. So I hope that Nick can turn on his camera and microphone. Nicolas Mantis is going to introduce his work that his laboratory, the Wharton Institute in the USA, has done on the understanding how the whole cell pertussis bacterial might be further characterized for potential future in vitro assays. So Nick, thank you for your time, and the floor is yours.
Sure. Thank you, Laura, for the introduction. So the project I'm going to talk about is very much of a research and development phase. It initiated as a collaboration with the University of Kansas. This is David Volkin's group, and formulating DTP and combination vaccines. My group at the Wadsworth Center, and Antigen Discovery, Joe Campo and his group at Antigen Discovery Incorporated in California. And the work was supported by the Bill and Melinda Gates Foundation. So I think everyone appreciates from the introductions you've already heard today that the Kendrick assay, also known as the mouse potency test, remains the gold standard for whole cell potency determination and batch release. And again, I think you also appreciate that it's a technically challenging assay in terms of both time, labor. There's some question as to its relevance related to immunity in humans. And I think the whole issue for today is also that it's incongruent with current animal welfare guidelines due to using both the large number of animals and relying on death as an endpoint. So again, the efforts, there are ongoing efforts to replace the Kendrick, focused on both serology-based assays, as Laura just introduced, and potentially moving to in vitro animal-free tests. So let me just give you a context of what the actual Kendrick assay involves. So it's a single intraperitoneal injection of mice. Over a course of 2-3 weeks, the animals develop an immune response, largely antibodies. They go on, and there's an intracerebral challenge, followed by survival, live dead, as a principal readout of potency. And I just want to underscore the theme of this talk will really be about, how little we actually know about the immune response associated with the vaccination. Oh, I'm sorry. Do people not see my slides?
I can see them.
I see them fine.
We can.
Okay, I just got somebody in the chat indicated they weren't seeing the slides. Yes, there's slides. So I'll continue. I don't know if somebody in the technical side can assist the person who's asking in the chat. So the question then is, so in the Kendrick's assay, that again, survival being the readout, you get a pass if the animals survive, and if Kendrick fails, the animals succumb. And so really the question is, what is the difference between an animal that was immunized and that survived and passed the Kendrick assay versus an animal that survived, was immunized and did not survive? And we really are focusing on the possibility that this all is leveraged based on the pivotal point here is the nature of the antibody response. And I guess the issue is it a question of the quality or the quantity of antibodies? And so from the literature and from the work we've been doing, we really think what's happening is that in a potent animal that receives a fully potent whole cell vaccine, they generate what we're calling high quality antibodies. And that's in quotations, as compared to an animal that receives a vaccine that's been altered, it's subpotent or somehow compromised, and those animals generate a low quality response. And from our data and others, the idea is that there's actually no difference in the total antibodies, but actually a question about the quality of the antibodies. And again, I'll continue, but I keep seeing comments in the chat about not being able to see the slides. Okay, I'll continue. So our research objectives was really, how can we think about moving forward to replacing a Kendrick assay? And our thought was it's really essential to better understand the nature of the antibody response and use that as a basis for further assay development. So our objectives are really, number one, determine what constitutes a high quality antibody response elicited by whole cell pertussis. Number two, what is the difference between what we're calling high quality versus low quality? And the relationship ultimately between a potent and subpotent whole cell pertussis formulation. Eventually, in step 3, we'd like to then use the information about what we learned between the high quality, low quality, and develop either serology and or in vitro assays to discriminate between potent and subpotent whole cell pertussis vaccines for vax release, validate these assays against the Kendrick, and ultimately move forward within the vaccine space. So what constitutes a high quality antibody response? And it's actually remarkable how little is known about the nature of the actual immune response to pertussis and in particular antibody response to pertussis. So we ended up employing a proteome array where we immunize mice with potent whole cell pertussis and then subject the immune sera to a HAMA1 proteome array to identify the antigens that are recognized upon vaccination. And our goal here was to really establish what we consider a high quality antibody profile response. And again, this was in collaboration with antigen discovery. And what you can see here then is just a heat map, and I won't go through all the details, but just suffice to say that each column is an individual animal. Each row is a different B pertussis antigen. And what we're comparing here on the left, 4 degrees would be a potent vaccine that we stored at 4 degrees prior to immunization. And the vehicle would be animals that received saline as a control. And the takeaway is the heat map, the color change is fold increase of antibody reactivity against a given antigen. So the question is, what are those antigens? And this is a list of the top 30 antigens that are recognized following whole cell vaccination in mice. And again, I won't go through all the details, but the takeaways are, in fact, that we identified 34 reactive antigens that were prevalent in greater than 10% of the animals. The top 8 antigens are outer membrane proteins. Virtually all these are associated with the bacterial membrane, and a large number of them are virulence factors and or associated with the acellular vaccine, and are also known to be reactive in individuals, particularly in animal models of the baboon model following actual pertussis infection. So the takeaway here is what we're getting is an interesting profile associated with the whole cell pertussis vaccine. The antigens that are being recognized are very relevant to pertussis infection and to vaccinology. So then the question is, can we sort out the difference between a high quality and a low quality response? And for this, we actually did what we consider a forced degradation, or we altered the vaccine through heat stress. So we took preparations of DTwP, subject them to increasing thermal stress, 60, 80, or 100 degrees for 1 hour, immunized animals, collected serum. We did basically the PSPT. We did a whole cell pertussis ELISA. We also performed Luminex using the list of antigens you can see on the right. These are largely associated with a cellular vaccine. And we also, these same animals were challenged intranasally with B pertussis 18323. And we looked at the ability of the vaccine to promote clearance. And so what we found is actually the thermal stress was in fact compromising the vaccine. So if you look in the left panel, this is intranasal challenge. So animals that received the 4 degree, just a single dose of the 4 degree stored vaccine had 10 to the 4 bacterial colony forming units in their lungs following challenge. Animals that had the compromised, the 100 degree vaccine, stress vaccine, had roughly 10-fold more bacteria in their lungs. And animals that were not immunized, sham immunized, had roughly 100 to 500 more colonies. So there's a clear dose response in terms of bacterial clearance following vaccination. If you do an ELISA, a whole cell pertussis ELISA, there was no difference in antibody titers across the different doses, different vaccine treatments. So animals that received the 4 degree, 60 degree, 80, or 100 all mounted very high titer anti-whole cell pertussis antibodies. But you do see distinct differences when you look at the antigen level. And this is just on the right, it's an example of pertactin. And this is a Luminex assay where we're looking at pertactin reactivity in animals. These are antibodies directed against pertactin in animals that receive the potent vaccine versus the thermally stressed. And there's a very definitive decline in antibody reactivity, suggesting that the stress resulted in degradation of the protectin in particular. So we then took those same serum samples from mice, again, that had received the potent vaccine that's stored at 4 degrees, 60, 80, 100, and we did the proteome array. And I think the takeaway is what we're seeing is very clear patterns of changes in overall immunoreactivity. And I'll just point out a couple of things. So in many instances, the vast majority of instances, the overall antibody response did not change across the different vaccines that were stressed. So an animal that got a vaccine at 4 degrees had a similar profile for many antigens compared to the animal who got the vaccine at 100. And that's, for example, in the purple, in BP 3755 at the top left, you can see that these are relative antibody units. And animals at 4 degrees have similar antibody binding to those that received the stressed vaccines. This contrasts with the one below that, BP 2497, where you can see at 4 degrees, the animals mount an antibody response in the green box. But as soon as you subject those vaccines to thermal stress, even 60 degrees for an hour, those antibody, there is really no reactivity against that antigen, suggesting that 2497 is a biomarker for thermal stress or stability associated, decline in stability or integrity of the pertussis vaccine. So that's one of our markers showing a decline in potency. By contrast, there's also the opposite, where 4 degrees with increasing heat, you actually see increasing amounts of antibody reactivity. In this case, we think the antigens are becoming more accessible or possibly even released from the whole cell pertussis. So again, these observations are consistent with there being sort of a high quality and low quality antibody response associated with potent and altered vaccines respectively. So how do we use this information then to think about moving forward beyond the Kendrick and thinking about a serology-based assay or even in vitro assay? So one scenario, so on the top again is just a depiction of the different models. So the Kendrick assay, again, as I've noted, involves a lethal endpoint shown in the top. The whole cell pertussis, as Laura introduced, is basically a vaccination serology, and then looking at antibodies, total antibodies against pertussis. And again, I think I've made the point that we don't see changes in whole cell pertussis antibody titers as a function of potency. So what we're proposing is one possibility is to modify the PSPT, where rather than coating with whole cell pertussis, you coat with particular stability or stress indicating antigens. So you can envision multi-antigens here. It could be protactin, it could be 2497, it could be other antigens, and that antibody profile by ELISA or Luminex would be indicative of vaccine potency. The alternative is, can we actually go to a cell-free assay that does not even require animals at all? And that concept would be shown here. So in this case, we generate high-quality reference serum. These could be either monoclonals or an immune serum. And then the setup would be a competition ELISA in which you coat your ELISA plate with B pertussis, particular antigens, indicator antigens. And then you do your competition with your different vaccine preparations. So a whole cell potent vaccine would be very effective at competing with the high quality antibodies, and you get very little reactivity on an ELISA plate. By contrast, a subpotent vaccine, which has loss or reduction in the number of relevant antigens on the surface, the antibodies would fail to recognize that bacteria, those bacterial particles, and actually bind to an ELISA plate. So the low binding versus high binding, in theory, would be sort of a measure of relative potency of that vaccine. And we only have very qualitative preliminary data to suggest that something like this might work. So again, this is a competition ELISA in which we're using DTP or W whole cell pertussis immune sera, and showing that a vaccine that's at 4 degrees is a good competitor of this serum in the setup that I mentioned previously. And when you stress that vaccine at 100 degrees for 1 hour, it's less effective. And that held true whether you use a whole cell pertussis immune serum hyperimmune serum or a DTwP immune serum. So again, these are qualitative, but they're again suggestive of the possibility that you can sort of probe the integrity of host cell pertussis using immune sera, and ideally a monoclonal antibodies. So that's where we're at. Again, I think we're developing these really in the research. These are the early days in sort of the research development phase. Ultimately, at some point, we need to benchmark these against the Kendrick assay and ultimately move forward, if possible, to the manufacturing space. So just to conclude, we've identified, again, what I think is sort of the first immune profile associated with host cell pertussis using our limited proteome array. There's clear differences in the antibody response between mice that received the potent versus a altered vaccine, again, thermal stress. And then our preliminary results suggest that either an antigen-specific serology-based assay may be able to discriminate between a potent and subpotent vaccines, and even this possibility, there is the possibility of moving towards a animal-free or even in vitro assay for potency determination. I'll just highlight that this work is in press. It's published. It's currently available on bioRxiv at DOI, but should be out in the ASM journal Microbial Spectrum within the month. And so with that, I'll just conclude and again highlight the individuals who are involved in this program and the initial support through the Bill Melinda Gates Foundation. So thank you.
Thank you very much, Nick, and apologies if the slides were not being seen by everyone. It will be available anyway afterwards, so thank you. In the interest of time, we move ahead and then we will take the conversations afterwards with the rest of the group. So thank you, Nick. And please, Heidi, the floor is yours for the last presentation.
Okay, thank you very much. I'm now having the pleasure to ask Dr. Sunil Goel to hold the next presentation. It's about manufacturers' approaches to implementations of updated DTwP testing strategies. Sunil is representing the DCVMN International, meaning the Developing Countries Manufacturers Network. Sunil, please take the floor. I'm happy to listen.
Thank you, Eddie. And it's a pleasure. Hi, everyone. It's a pleasure for me to be here at HSI and AFSAC Collaborative Forum to discuss with you manufacturers' approaches to implementations of updated DTwP testing strategies. Next, please. Yeah, thank you, Laura, for your support. This presentation provides a brief overview relating to implementation of updated testing strategies for DTwP vaccines, testing at manufacturer level, that example will be Serum Institute of India, at country level, that is India, at regional level, that is developing countries vaccine manufacturers network, and global levels. And this presentation identifies the test of most concern with implementation of 3 R's, that is refinement of method, reduction, and replacement. And this presentation also shows how the positive intention of implementation of approaches in collaboration with regulatory authorities could serve as a powerful tool in reducing animal use and suffering. Next, please. Serum Institute of India is committed to the development, introduction, validation, and implementation of 3 R's and on consistency-based approaches. It has been a front runner in implementation of 3 R's since 2003, even when regulatory guidelines did not recommend any alternative. And this was with support of NRA, NCL, and Indian Pharmacopoeia, which had been always supportive and receptive to such initiatives. Next, please. Serum Institute of India had strong belief in consistency approach, and based on which alternative methods were used for characterization of vaccine along with in vivo methods. Suitable correlations were developed and monitored for number of batches as huge as 200 lots. More emphasis was given on data monitoring of critical parameters and trend analysis, which eventually led to successful implementation of non-animal methods without compromising the product quality. Next, please. At Serum Institute of India, 3 R's has been successfully implemented in not many areas, but our main focus for discussion will be DT-based combination vaccines. But in addition, it has also been implemented in rabies, hepatitis, conjugate vaccines, IPV, and it has been 100% deletion in all the vaccines with reference to abnormal toxicity test and rabbit pathogen test, which has been replaced with BET. And further advancements are also in progress. Next, please. Coming over to the scenario in the country, that is India, we have identified that manufacturers have active 3Rs programs, and that is because of a lot of collaborative studies, lot of involvement of various agencies who have been pushing up the implementation of 3Rs. And it is good to see that we have implemented more than 3Rs opportunity for batch lists like single dilution assay for DNT, replacement of rabbit pathogen test, in vitro potency for hepatitis A and B, deletion of abnormal toxicity test, which is now complete. And it is also good to see that manufacturers are now committed to invest in improving their 3Rs program, including the transition to in vitro potency test for DTAP and to find a replacement strategy for whole cell pertussis vaccine. Next, please. And how is this possible in the scenario in country? Just to tell you that the National Regulatory Authority, the National Control Lab, and the Pharmacopoeia Commission jointly has a meeting with vaccine manufacturers at regular periodic intervals and discuss various issues there. And one of them is implementation of 3Rs, and which has been proved very successful in implementing a new method, which Pharmacopoeia also goes ahead with publication with full support. For an example, Indian Pharmacopoeia has constituted experts group working group on implementation of 3Rs. It has published chapters on substitution of animal assays with in vitro assays, that is general chapter on alternative methods, replacement of rabbit pathogen test with in vitro assays, and now it is also in the process of publishing a chapter on next gen sequencing. And this would not have been possible without the support of NRA and NCL, which are quite open for discussion, and also we can see their willingness in implementation of 3Rs, and the procedure gets expedited soon with a faster review and imparting suggestions wherever required, and finally the approvals are granted. Next, please. Coming over from the country scenario to the development countries manufacturers network. Again, we have observed increased number of manufacturers are now having active 3Rs programs and with the 3Rs methods implemented. For example, single dose, single dilution for DT based combination vaccines, deletion of abnormal toxicity test, and commonly I mentioned in vitro potency test for hepatitis A and B and for rabies as well, D-antigen for IPV, and replacement for rabbit pathogen test. But one difference we could observe is there are different approaches in prioritizing replacement and other 3Rs in different regions. One region has complete thorough scheme for implementation, other region does not have any implementation procedures in place. Hence, there is a need to align locally, regionally, and globally on further investment for method development and implementation, and on sharing expertise and experience on available methods for other animal tests as well. As we have seen various scenarios from MHRA, CNCNO, and other forums who have been contributing great towards this, and we also look forward for collaborating with these agencies for implementing in vitro assays for DNT combination vaccines. Next, please. We have identified certain areas where we see certain opportunities to be implemented and to be explored. First of all is single dose, single dilution for DD based vaccines to fill the gap until and unless we have in vitro assays in place, validated and available in regulatory guidelines. Second is replacement or deletion of specific toxicity test, in vitro potency test for DT, acellular and whole cell pertussis potency by in vitro method, whereas we see whole cell pertussis refinement for short term, but finally replacement as a long term strategy. And the dream would be to combine in vitro potency assay for DTAP or DTwP in one single assay like multiplex assay shared earlier. In addition to this, we also look forward for changes in other assays like rabies in vitro potency, which EDQM BSP 148 is going ahead fast. Recombinant factor C, which is also moving fast. European pharmacopoeia has already led to deletion of rabbit parogen test and BT replacing with recombinant factor C and monocyte activation test. And we also see high throughput sequencing coming up as a major issue. And then finally, abnormal toxicity deletion for non-indigenous market is another target which we are focusing upon. Next, please. Here in this case, I wouldn't like to speak much because my colleague Pradeep and Morgan has already said a lot about it. That is replacement of multiple dilution challenge assays with single dilution, single dose assay for DNT potency. Sorry, next please. We can skip further. Yeah, next please. Yes, what do we achieve? We implement reduction, that's 1R, that is from 112 guinea pigs for each, that is 224 guinea pigs we require for testing of diphtheria and tetanus component having been reduced only to 30 guinea pigs when we implement single dose serological assay. That is 85% reduction in animal consumption. Next please. And we also see the implementation of refinement. In pertussis challenge assays, we see that we eliminate the distress and trauma in animals by refining the method. And definitely it leads to a manufacturer's advantage, consumption of less animals, which leads to cost effectiveness, maybe helping in reduction of cost of vaccines, and also availability of space in animal house can be allocated for other assays, and it also leads to testing of more batches in existing capacity of animal house. So that's the benefit of implementing single dose, single dilution method. Next, please. Now coming over to wholesale potency, wholesale pertussis potency and DT in vitro potency. Laura, do you advise me to speak on this? I think so. You have already spoken most what I wanted to speak here. Anyway, I'll just say some few words. Yes, Laura, please.
No, please go ahead. Share your point of view, Sunil, please.
Okay. So the DCBN project on pertussis serological potency test was a good success, which eventually collaborated with vaccine manufacturers as well as national control laboratories of some countries, and we could generate some data, which the report is in publication in WHO forum, and it is found that it will work further with improvement to the methodologies carried out. Then coming over to wholesale pertussis in vitro potency, method is not available, but we are looking forward for the earlier discussion which Nicholas has just discussed, looking forward to that. And then we have discussion on best strategy to be considered. Short term, like we see 5 years down the line, we can see the new strategy for in vitro tests to come up. Till that time, should we go ahead with PSPT or should we continue with mouse Kendrick's test? That's another strategy to be designed. And then DTAP in vitro potency. It is good to see that some DCBMN manufacturers are in the process to test the methodologies developed within VAC2VAC. That's another positive point. Next, please. But there are certain challenges to phase out these animal testing in DT-based vaccines, which is categorized into two. First is technical, like challenging characterization of legacy products. Age-old products like DTP vaccine is one of the technical challenge which we feel. Then there is lack of experience on product specific validations to be performed by the manufacturers, and then method to be transferred to the NCLs releasing the product. Another, which Paul also mentioned, reagents availability has to be very much available to the manufacturers as well as NCLs, and the role of international reference standards in validation studies needs to be evaluated, as there is no one-to-one correlation between in vitro and in vivo assays. However, different GMP quality, and regulatory compliance maturity of manufacturers, NCLs, and national regulatory agencies is also one of the challenges which requires to be mitigated. Coming over to the regulatory, lack of knowledge, experience, and confidence on new assays and data analysis, slow pace in changing local regulations, pharmacopoeias, different standards and lack of alignment, and finally, strong risk aversion mindset and other non-technical biases of policymakers and NRSAs is another challenge that needs to be discussed. Next, please. But the journey has already started. We thank VAC2VAC, DCVM and PSPT project, EDQM, BSPs, PATH, NIAMBL, who have been successful in demonstrating the concrete opportunities to use methodologies to replace, reduce, and refine animal testing. And we also thank HSI, EFSA, NC3Rs, etc., multiple stakeholders, which they are taking forward the outcome of the earlier said agencies. And finally, the updated guidelines and registrations in many countries, including the developing countries, has also started happening. So we look forward for many such more positive outcomes, which will definitely help us in implementation of 3Rs specific in DTwP group of vaccines. And the last slide focuses on, next please. What are the needs to carry out this change? First of all, there is a need for investment on trainings. Lots of the vaccine manufacturers in developing countries and NCLs, they demand a lot of trainings for implementation of these changes. There is a need of sharing SOPs or facilitated method transfer between the agencies who have collaborated to the other counterparts, or between the agencies like VAC2VAC, if they have some SOPs that can be transferred to other agencies, shall be given a boost. Then there should be improvement in supply of reagents and new technologies at affordable price. One example is monocyte activation test. That technology available from the market is a very high cost involved, which gives a resistance to take up that for implementation as a change for rapid pathogen test. Then there are a lot of complexities related to vaccine manufacturer structures. Some are privately owned who can afford to buy new technology, but there are certain public institutes who have to follow the government procedures for implementation. Then there are local legislation challenges, and the way regulators and pharmacopoeias operate needs also to be taken care of. Then there is a need to promote early dialogue between the manufacturers and regulators on non-animal testing strategies for novel products and legacy ones. As an example, Serum did in 2003, 4, 5, when none of the pharmacopoeias had published or other guidelines had published this method for implementation of single dose, single dilution challenges. Then there is a need for agreement on data-driven planning and on data sharing with regulators. And last but not the least is need to kickstart new local or international projects or new business opportunities, like projects like just Paul discussed with Catherine. That's another welcome. And similarly, we look forward for other collaborative studies as well. Next, please. Thank you very much for your kind attention.
Thank you very much, Sunil, for the presentation full of very important information. What is happening within the DCVMN? Thank you so much.
My pleasure.
Thank you, Sunil, for my side as well. So thank you very much for all of you for sticking with us on this journey to introduce you the opportunities for the potency. We still have a few minutes, so I would like to see if all the speakers can come back with their videos, and if we can just really maybe getting some remarks about, in general, what is your perspective on the importance of strategic planning? So many of you have shared the experience that you had in implementing from the single dilutions to the in vitro potency. We have been seeing that there is the possibility also on the whole cell pertussis that of course needs to further develop, but it would be interesting to gather your ideas about if you are a regulator or a company or a national control laboratories, what are the key considerations that you would have in considering what is the best testing strategy that should be taken up or implemented? So again, knowing that many of the developing countries stakeholders might be online, and some of them, they already started to think about these strategic changes that they need to make in their testing strategy. If you have any kind of thoughts, recommendations, or consideration in the way they are approaching this important transformation.
Yeah, Laura, from manufacturer's point of view, we are always guided and surrounded by regulatory guidelines. We may have new method developed, new method validated, new method implemented as well. But until and unless the regulatory guidelines support a manufacturer, even the NCLs, they follow the regulatory guidelines point by point. So that is another area of concern. We do get support from our regulatory authorities, but in some countries where their law of the land is so stringent, they do not permit implementation of new methods or new assays until they are convinced or until they have personally observed that assay. That is one area I would like to propose for discussion.
Thank you, Sunil. And I would say that probably the approaches that the VAC2VAC project had to engage the regulators during the whole process of the method development probably was also one of the key successes, and also a little bit of what we try to do within the DTwP and PSPT to also have the NCL laboratories to work together with the manufacturers on the assessment of the serology might be maybe an approach to hopefully create a little bit more confidence on the possibility of the change. But again, that's what we were able to see today through those experiences. But really, I'm happy to hear from the others if they have any other considerations or comments. If I may take the case of the single dilution, because there were some interesting exchange in the chat. This is something that is very interesting in terms of the strategy that a company might be taking up on whether to consider the investment on the single dilution versus considering some other opportunities, even if those are probably yet to be well defined, and there is the investment on the method validation that still need to be done. But we have been seeing how the consistency approach and the data that you have about your project might actually give you the answer about whether you should prioritize as a short-term the single dilution versus perhaps if you don't have strong data to identify what is the best single dilution that you can choose, then perhaps that is not a strategy that you can consider. But again, open to the opinions and thoughts of the others here.
I mean, I can comment a little bit on that, or at least share a view. I think if someone is already doing a multiple dilution assay and that's being used routinely, then provided you can fulfill the requirements of implementing a single dilution assay, it's just it's a maths exercise. Okay, I don't want to ignore the fact that it requires a lot of engagement with authorities and paperwork. Of course it does, but it's essentially a paper exercise, right? that's where it can be done. It can't be done for all products and all components because there are certain prerequisites you have to be able to meet, and that just can't be done in some cases. But where it can be done, I think it makes sense to explore it. But it's still then, even though it can provide ongoing demonstration of compliance, doesn't tell you an awful lot more. You don't get any kind of quantitative measure. It tells you nothing about consistency. It still uses animals and has all of the flaws and the issues related to that. So for me, it's potentially a stop along the way, but it isn't the destination that we should be hoping to arrive at. Thank you.
Thank you, Paul. Please, Silvio.
Thank you. If I can make an example of why authorities insisted for one of our products to move from traditional readouts in international units per single human dose to consistency of production. Our X-Exim hexavalent product, as you have seen in my slide, is combined with, there is a morphilus in it, is a polysaccharide. It's conjugated to a tetanus carrier. So historically, of course, our products were always conform to the 40 IU per single human dose specification, but our X-Exim product, because of this double tetanus component, often was very high. You know, we had very high tetanus responses, right? And that's where it was the interesting point of view of the health authorities. They say to us, you know, I know you will always pass the specification by design. However, what I want to see is that you have a value that is consistent with your manufacturing. I want you to possibly write off something that is still potent, over 40 IU per single human dose, but is not consistent with what you have been able to demonstrate so far for your tetanus response in terms of antibody content. And that was a very interesting point of view that didn't come from us. We were more conservative in the way of thinking, but that, of course, with another perspective. So basically, please set an acceptance limit for your product and for your consistency of manufacturing. And I think that is, very interesting in this new perspective of the consistency approach. But we already seen how important is to ensure, of course, that we are not releasing potent products or whatsoever. And also, it's been very interesting the discussion we had about flocculation and how flocculation intervene in the vaccine formulation. But let's say, with the hope for the future, nothing is now replaceable. It's just the quality of the data. I think we managed to gather to sustain and continually sustain the change that could help us, you know, go forward with the 3 R's maybe.
Thank you, Silvio. There are any other comments? What we have been seeing that, again, our strategies might really need to be tailored to the products, and there is probably the need for the companies also to be at the forefront and be the ones that are trying the available methodologies and again, try to share as much as possible their experience. From our end, thinking about going back to the wholesale pertussis, that is actually our open challenge here. We will definitely need to have this, not only to invest a lot in the basic understanding of the bacterial itself, but at the same time, it will be very important to hear from the companies and the regulators that are still actually using the traditional methodologies in order to understand exactly what are their expectations and making sure that the way we hopefully eventually get into the development of a full in vitro will actually leverage all the important lessons learned that have been shared today on the path that has been followed so far for the definition of the in vitro potency for tetanus, diphtheria, and cellular pertussis. It will be very important for us to be a partner with all of you, making sure that the regulatory authorities, including WHO, is going to be our stakeholder and is going to be included into the future conversation, because whether it's going to be the in vitro for tetanus, diphtheria, and the cellular pertussis or the whole cell, we need to make sure that all the key elements about the method validation and the regulatory acceptance are in place, including the reagents, including having access to the different methodology, but at the same time, also being aware that there might be the work that is coming from the different manufacturers on identify how those methodologies can be correctly implemented and optimized for their own products. This is something that I hope that it's a message that we can take back home, because a lot of important work it has been done, but now the next piece is really making sure on how those methodologies can really be implemented for the specific product. So hopefully we gave you an idea about that this is possible. Of course, a lot of investment and work is needed, but at the same time, we are all here to make sure that if you need access to expertise or if you have any doubts about what we have been shared, we are here, and you can get in touch with all of us, and we can work together. If not under an official umbrella of a collaborative study, but there is opportunities like this to share thoughts, ideas, concerns, or also successful case studies. I don't know if there are any final remarks from the others. Otherwise, I'm actually happy to thank you very much for being with us, for the time that you spent. Your questions are very important also for us to understand a little bit your perspective and issue. And as mentioned, the recordings and all the presentation will be available shortly in the event page. So again, looking forward to see you again in the next event. So we are probably having another one dedicated to the neurovirulence. And then please save the date, because together with the International Alliance for Biological Standardization, there will be a final conference dedicated to the in vitro to in vivo transition for both human and veterinary products in Bangkok in December. So first week of December. So a save the date will be shared very soon with an additional agenda. So we hope to see you in person in Bangkok at the end of the year. So again, thank you very much to the speakers, the members of the scientific committee, and to all of you for being with us yesterday and today. Thank you.
Thanks, Laura.
Thank you.
Thank you, Laura.
Goodbye.
Thank you, everyone.
Thank you. Bye-bye.